All right. And so now I am going to press the record button.
Lisa, just out of curiosity, I'm on the YouTube channel for TCBS, but I don't see a live stream thing.
Okay. Well, let me.
Do I have to subscribe?
No,
I
no.
can see it.
Maybe try refresh.
Okay, got it.
Awesome. Oh, yeah. Right, right. Yeah. Sure. Because too much going on for you.
I mean, I don't need the Zoom. I'm not going to Zoom.
Oh, yeah, yeah. You can just watch it on YouTube if you want to.
But I guess, do I need to?
But
Oh,
if
no,
you
I
want-
need the Zoom.
Because you need to talk to me, yeah.
Okay. All right. I still might come in here and do this.
Okay.
When are we doing this?
Like right after the line acknowledgement, so like in 5 minutes.
Yeah, okay, let's just do this first.
Okay.
I'll just read this.
Okay. You want to come in now? Or you want to go?
I'll do this right back.
Okay.
Jordan, check your chat.
Trying to remember.
Well, good morning, everybody. I am so excited to welcome you all today to our professional internship presentation event. This is with the University of Hawaii at Hilo, Tropical Conservation Biology and Environmental Science Master of Science program. And to begin our morning, to open our morning, we're going to have an ole that's going to be presented by our own Kavehi Lopez and Carmelita Villalobos. So I will turn it over to you two.
E ho mai ka ike mai luna mai e o nā mea huna no e au o nā mele. E ho mai, e ho mai, e ho mai e ho mai ka ike mai luna mai e. O nā mea huna no e au, o nā mele. E ho mai, e ho mai, e ho mai e ho mai ka ike mai luna mai e. O nā mea huna no e au, o nā mele. E ho mai, e ho mai, e ho mai e.
So next we just want to give a land acknowledgement. Here at UH Hilo, and especially at the start of the TCBES program, it was very important for us to acknowledge the spaces that we are doing our work in. So we just want to do that again today, and respect the land and the place that we're coming into and working on.
Aloha nui kako and mahalo nui for joining us today for the TCBES Professional Internship Symposium. We'll be starting off with a land acknowledgement in an effort to honor Aina and Kanaka Oiwi, the indigenous people of Hawaii. As we gather in this virtual space, we recognize that the places that we work and live in have been severely impacted by settler colonialism. We also recognize that Hawaii and its people have what Hawaii and its people have endured as an illegally occupied nation. This intimate connection between Kanaka and Aina was taken by force. Land dispossession, as well as the loss of language, culture, and spirituality, continues to fuel the intergenerational trauma experienced by Kanaka Oiwi across the Pai Aina. We must all acknowledge this harsh truth and move forward towards building trust, relationship, and allyship with the Kanaka Oiwi of this land, as they are inherently tied to Aina. The sustainable future of both is dependent on this undeniable familial bond. As scientists within the TCBES program, we acknowledge our kuleana to take the steps towards restoring this relationship between Kanaka and Aina. Mahalo.
Thank you very much for that powerful land acknowledgement. Thank you, Carmelita and Kavehi. I now would like to invite the TCBES directors, Tracy Wigner and Becky Ostertag, to join the presentation.
Oops. Aloha, aloha, I komo mai. As Director of the Tropical Conservation Biology and Environmental Science Graduate Program at the University of Hawaii at Hilo, I am very pleased to welcome you to Spring 2021 UH Hilo TCBES Professional Internship Presentation Day. Before we begin with our extraordinary program, I would like to tell you a little bit about the TCBES program. Our mission is to provide Masters of Science level graduate education that fosters the knowledge and techniques in conservation biology, and environmental sciences, including basic, applied, and socio-ecological research, and promotes scholarly activities in marine and terrestrial environments that will enable participants to pursue careers in research and natural resource management. The program utilizes the extraordinary biological, physical, and cultural complexity of the island of Hawaii as a focus of investigation and study. Graduates of the program are highly successful at obtaining technical positions as scientists, resource managers, and educators, and admission into PhD programs in related fields. The program began in 2004, and we've graduated more than 200 students. We currently have 45 faculty associated with the program across 5 different colleges. And we have 53 active students, but not for long. 10 will be graduating this semester. And we're very excited about that. Join us again on May 14th for our Zoom-based graduation ceremony.
Okay, in the fall 2018, we expanded our internship track into a professional master's degree. The students here today conducted more than 600 hours of internship work in an immersive experience. In the past, the majority of Hilo TCBES students followed the thesis track, received an excellent training in research, and then many of the graduates went on to work in natural resource management rather than continuing their education and obtaining a doctorate. With this reality in mind, Hilo revamped the internship track option to align to courses with knowledge and experiences that a graduate needs to actively contribute as a scientific professional in an environmental and conservation agency, NGO, or other organization. Part of this revised curriculum is a rigorous internship project. And now, more than ever, we need science to inform decisions we make about conserving biodiversity, about the management of natural resources, and we need social science to understand how humans make decisions that affect the well-being of our planet. We need to understand the human-environment interactions. And that's the advantage of Hawaii Island. We have something here that's truly special, unique, a spirit of aloha and a collaboration that we can see so clearly in the audience, all of you that are listening right now, with Hilo students and faculty, with partners, nonprofits, from state and federal agencies, from seasoned veterans to those just starting out in their career path. There's a sense that we can and will do this work together. In this small island system, we collaborate because we have to, but also because we enjoy and we respect each other. And we know that's what's Pono, because we have this beautiful wisdom of the indigenous knowledge on which to build. So we invite you to listen today to our 6 featured speakers. They're shown here. This picture was taken in their first semester when they take a core class all together. And they, in this class, they do field work and learn how to do science in the Kipuka of Hawaii Island. We invite you to listen to them today, to observe, to question, to critique, and to share knowledge in this event. Welcome, and we look forward to learning together.
Aloha, everyone. So I have the pleasure of welcoming our very first mentor. I think, though, we might be a wee bit ahead of schedule. So what I'd like to do is to make sure that everyone is on the same page time-wise, because people are going to be coming from across the US to join us. We're going to go ahead and we're going to take a 6-minute break, and we'll return at 8:45 to hear our first presentation. So thank you all for joining us this morning for our opening, and we will see you all at 8:45. All right, thank you all for joining us and welcome back. And thank you, our directors, Tracy and Becky, for setting the stage for today's main event, the Professional Internship Program. I would like to kick off this morning by introducing you all to one of Jordan Anderson's mentors, Dr. Carla McDermott. Dr. McDermott is a professor in the Department of Marine Science at the University of Hawaii at Hilo, our very own home. Carla is famous for her love of limu and her expertise in the field. Her passion for marine science and science education is contagious. She has mentored many TCBES graduate students over the years, both through thesis and internship routes. Please join me in welcoming Dr. McDermott to the Zoom microphone.
Thank you, Lisa, but that was too long an introduction. Today is really to focus on the students. And before I start, we're going to turn you first talk to the ocean. So next year, I want to hear not just a land acknowledgement, but also an ocean acknowledgement. And Jordan will do that. She did that through her research. I first met Jordan in the fall of 2019 when she had just arrived from Manoa. And she said she was interested in limu. And I said, great, I need somebody to help me teach a workshop in a couple weeks on seaweed identification and hands-on with specimens and microscopes. And she said, great. So I took her. I didn't know her. I didn't know if she even knew what a species was. And she did great. She was a great teacher. She was fearless. And oh, by the way, it was fourth graders. Then she decided she wanted to work on a project to combine not just working for an agency, but she wanted to do some of her own experiments. And she has created a very interesting professional internship. She worked with Ocean Era, and I see that her mentor, Keely Martin, is also here supporting her. Thank you, Keely. This semester, I was lucky to have Jordan as a teaching assistant, well, really a partner, a team teacher in a lecture class, which is unusual for a graduate student instead of just a lab class. But I thought that it would further her experience and her career. And my goodness, she is a quick study. She's organized, she's fun, she's caring. The students love her. She has a command for the information, and she taught me an awful lot. And I personally am going to be sad to see her go. The department is sad to lose her, but we are also so happy that she has completed her master's. And so, Jordan, I know you're eager to share with us your knowledge of the ocean and of the seaweeds. Take it away.
Thank you so much for those kind words and that lovely introduction. I'm going to share my screen. I get a quick thumbs up that looks okay, Lisa. You're looking good? All right. So thank you again, Carla, for that introduction. I wouldn't have wanted anyone else to introduce me here today. Aloha to everybody else. Thank you all so much for being here. Before I get started, I do just want to say thank you to Carm and Kevehi for presenting our Oli this morning, and also the land acknowledgement, which we'll add the ocean in eventually. Thank you to Lisa and Becky and Tracy for setting the stage this morning and introducing our program. I'm really honored to be starting us off with the presentations this morning. As you've heard, my name is Jordan Anderson, and I'm here today to tell you all about my internship experience here in the Tropical Conservation Biology and Environmental Science program. I want to congratulate my cohort members, the ones presenting with me here today, as well as the ones that will have the pleasure of hearing this upcoming fall. I couldn't be more proud of you all. Now, the focus of today's talk. This titled Assessing the Reattachment Potential and Large-Scale Aquaculture Feasibility of Native Hawaiian Macroalgae. Some of the species that I worked with, both in my experiments and at Oceanera, which is my internship agency, are pictured here. On the left here, we have a fragment of Amansia glomerata, which shows some really interesting results in my reattachment experiments. And the two right-hand photos are of Halimenia here, and then myself holding some Halimenia, one of the species grown at Oceanera that I can just never seem to take my eyes off of. I think it's one of the most beautiful plants. I have a pressing of it hanging right above me as I present here today. This work would not have been possible without the support of my committee, Ms. Lisa Canale, the internship organizer, Dr. Carla McDermott, who oversaw my experimentation work, and of course, Keely Martin, my mentor at my internship agency. And I'll talk about that in just a few slides. So speaking of those next slides, here's what you can expect from today's presentation. To start, I'm going to first bring us all up to speed on Native Hawaiian algae, or do the best that I can in the limited time that we have here today. Once we're familiar with the plants that I had the fortune of working with, I'm going to walk you through the experiments that I conducted to explore the reattachment potential of different species of native algae. To conclude, I'll introduce you to the organization and share with you my time at Oceanera, where I not only learned practical aquaculture skills, such as caring for species of red algae, like a Gardiella, which is pictured here, but also practiced what I like to call the art of writing standard operating procedures for a living, working aquaculture facility. And it wouldn't be my time as a graduate student without sharing with you quickly my time as a teaching assistant. So, to understand why research on native algae is so important, it's key that you understand why these species are at risk. Unfortunately, there are a suite of threats that are mounting against many species of algae within the Hawaiian Islands, and really around the world for that matter. There's no doubt that ocean conditions are changing. Temperature and acidity are on the rise at global scales, and at a more localized scale like we see here in Hawaii, conditions are changing even more drastically due to high inputs of nutrients from sources such as runoff and sewage. This photo here in the bottom right was taken off the coast of Maui, where inputs of nutrients are incredibly high due to sewage and cesspool drainage. This is also a problem that we see really prevalently here on Hawaii Island. You can see this cloudiness or murkiness in the water, and that is actually the seeping of these high nutrient waters through the bottom of the ocean and into this nearshore reef environment. Increased nutrient loads are not alone in their siege against native species, as pollution also proves to be a formidable foe. Camilo Beach is famous worldwide for its accumulation of physical pollutants, and we find that right here on our own island, pictured here in the bottom left. I'm sure Mike will tell you all about Camilo Beach in the fall. So while trash is not consumable to algae, it still poses a threat to the ecosystems that they are a vital part of. What I would call the most dangerous threat to native species here in Hawaii is invasive species. Just like Kavehi is going to tell us about later on this afternoon of the terrestrial threat of invasive species, there's also invasive species of algae that are here, and they unfortunately outcompete natives. In the top right photo is Kaneohe Bay over on Oahu, where species such as Capophycus and Yucuma have taken over entire reefs and are fittingly called the smothering seaweeds. Fortunately, those two species aren't commonly found here on Hawaii Island, but there are a suite of other invasives that pose a huge threat to the native assemblages of algae, which our reefs hold, and they help to keep in place a really delicate balance that the ecosystems have. If you know why algae species are threatened, it's also important that you know why it matters that we protect these species, why we conserve them. Broken down to the three simplest descriptions of why native Hawaiian algae are important. We note that 1, they're a food source, 2, they hold cultural significance, and 3, at their most basal level, they still are primary producers. You might not realize this, but algae account for producing half of the world's oxygen. So for every other breath that you take, you have an algae to thank for it. But let's pretend for a moment that providing us with the most necessary element on Earth, that being oxygen, let's pretend that wasn't enough for these species. Why else do we care? Well, here in Hawaii, not only are these native species an important food source for reef fishes, as well as our honu, the green sea turtle, which we all know and love, many of these species are also an important food source for the people of Hawaii today, and they have been especially important historically. In addition to being a food staple to the people, some of these species also hold significant places in the culture to represent forgiveness, such as limukala or a sargassum species. These plants have great healing abilities, not only of physical wounds, but also emotional ones. And they're given to a party who has been wronged, and then the two parties eat them together as a sign of reconciliation. So we could talk really all day about the reasons why, but I hope for just these few that I outlined here that you can see why native Hawaiian algae or limu, why they're so important. So with that quick background of not only why algae are important, but what threats they face today, let's jump into the focus of my internship projects. If you split my internship experience in half, here is the first half. And that was completing an experiment with Dr. McDermott of the Marine Science Department as my mentor. We brainstormed for what I felt like was hours on what would be an important question to ask for my master's internship. And we actually decided to build on the work of a previous tropical conservation, biology, and environmental science student, Miss Cecile Walsh, and further explore the reattachment potential of native Hawaiian algae. Miss Walsh did this back in 2014 with a thesis that was titled Reattachment and Transplantation of Native Hawaiian Macroalgae, a potential tool for reef management and alien algae mitigation. In that, she tested 22 different species of algae to better understand their reattachment potential. So, building off of this work led me to my primary question, and that was, does fragment size impact the likelihood of reattachment in 5 different species of native Hawaiian algae? As you'll see in the coming slides, my project expanded to explore more than the originally decided 5, but for the purpose of this question and purpose of the results, we're going to focus on those 5. The leading null hypothesis would be that there is not an impact on the ability to reattach because of the fragment size for any of those species. So, the species that were included in my experiments can all be seen here. Something you should know about algae is that they can be divided into one of 3 phylum. Phylums being a major taxonomic grouping, so large that humans and cats actually belong to the same phylum, right? So very large groupings. So with algae, there's 3 major phyla. There's the rhodophyta, which are the reds, the chlorophyta, which are the greens, and the phaeophyta, which are the browns. And in this experiment, we made sure to include at least one representative from each of those 3 groupings. So given their names, you may have been able to deduce from these original 5 species, which are on the left. My red representative is pictured farthest to the left here. This is Pterocladiella capiculi. The top of my second column here is my second red representative, which is Amansia glomerata. In the bottom of the second column is my phaeophyte, or my brown representative, being Podina. My green algae were represented by 2 species. At the top of the third column, we have Caulerpa lentilifera, and the bottom of that same column here is Microdictyon, which is a really cool, really net-like green algae. Also photographed here are the 2 species that I worked with that were grown over at Oceanera. They were included in my study, but not for the entire duration. The species are Halimenia, which is at the bottom of this column here, and then Agardiella, which is all the way on the right. So I could spend my entire allotted time here walking you through how I did these experiments, but instead we're just going to go through the most basic steps. So most of the plants that were used in this experiment were collections from the wild. I did them myself. They are from either Richardson's Ocean Park or Onekahakaha. Both really popular snorkeling spots here in Hilo. If you're on island, you probably know where they are. I would just like to note that when I collected these plants, I collected them as sustainably as possible, in that I took only the plants that were needed for my experiment, and when I harvested them, the holdfast was left behind. If you're not familiar, the holdfast is basically the algae's equivalent of a root system that we see in a terrestrial plant, so it allows them to attach to the substrate and grow. So when you collect a plant and leave behind the holdfast, the plant has a chance to regrow from the same point. So that occurred for all of my wild collections. Species that were not collected from the wild came from one of 2 aquaculture facilities. As I mentioned previously, there was 2 species from Oceanaria that got added during my experiments, Halimenia and Agardiella. The third being Caulerpa lentilifera, which was the final aquaculture plant that was provided to me after being provided to Dr. McDermott from the Pacific Aquaculture and Coastal Resources Center, or PACRAC, which is the aquaculture facility right here in Hilo. Once these plants were collected, they were given 24 hours to acclimate to the experimental conditions, which were set up in 5-gallon buckets with a single air stone and a covering of plastic wrap. The plastic wrap was not actually initially in the experimental protocol, but it was added to keep out bugs and lizards, but it only did part of its job. We did this experiment during prime termite time here in Hilo, and I came in one day with an entire layer of termites covering my buckets. So we went along and added those. I also mentioned lizards. I did have 2 lizards that got into these buckets and died. And while I don't necessarily have the data support it, I do think they impacted my experiments. But regardless, after the 24 hours of acclimation in the buckets, the plants were divided into their appropriate size categories. They were attached via raffia, which is a natural fiber, 2 pieces of rubble that was coral. So the topmost photo here shows myself tying a piece of Caulerpa, that green algae, to the rubble by wrapping the raffia around twice and creating an X-shaped pattern over the plant. And that was standard to attaching all of the plants to their pieces of rubble. These were then set into the buckets. The air stone was put in the middle and it was turned on, and they were left outside the Wentworth building on the UH Hilo campus. The bottom right photo is the standard setup for these buckets with the 6 randomized pieces of algae attached to the substrate around the edge, with a single air stone in the middle, which was weighed down by a rock so that it didn't float to the surface. They were left there for a total of 3 weeks, and at every week the water was changed and the buckets were completely cleaned out. After the 3-week period, the raffia was removed by cutting it on the opposite side that the algae was attached to, and then carefully removing that raffia. And then after what I'm calling a gentle yet firm shake, one quick wrist movement, if the algae was still attached to the rubble, it was marked as reattached. If it did not remain, then it was marked as not attached. Some of the plants attached extremely well, others not at all. They didn't even dry. And then there were those that were kind of an odd case, just like Amansia that I mentioned in my first slide, which actually did the strangest thing and reattached to the raffia fiber itself rather than the rubble substrate. So let's take a closer look at some of those results. I'm going to orient you first to this figure. So on the y-axis, or the vertical axis, we have the number of samples. And for this figure, the horizontal axis, or the x-axis, shows the size category of the algae. And this is for all of the species that I used grouped together. In green, or in the mint color, we have the number of plants for each size category that did not reattach. And in blue, we have the number that did reattach. So what this figure displays is actually that the experiment showed us that size does not impact the likelihood of a species to reattach. The statistical analysis that I did were chi-squared tables using Minitab 2020 software. They supported this conclusion, but I did not include the numbers here in the figure. If the species were going to reattach, they were going to do it regardless of size. But as you can see with this second figure, where now the x-axis has changed to species, not all of the species actually reattached successfully. The colors represent the same thing. So green means it did not reattach, blue means it did, and our y-axis still remains sample size. And while you can see here, the sample sizes are a little uneven due to the difficulty of consistently finding some of these plants, which is just one of the realities that I learned doing these experiments. Seeing that not all of these species consistently reattached is actually pretty interesting to me, because all of these species were chosen because they all had successfully reattached in Miss Walsh's previous work that I based these experiments off of. So while I recognize there are many things about our two experiments that were different, I think it just goes to show how much still needs to be uncovered about the reattachment process in algae. This may not have been what I was expecting. There's still a lot of value and implication in these results. So seeing that, you may be asking yourself, what does this mean? Jordan is here telling me it's important, but why is it important that we understand how algae fragments reattach? Why they reattach to pieces of rock or pieces of string, and what are the next steps in this knowledge? One of those future steps could be ensuring that outplanting is as successful as possible when it's implemented. Reattachment of algae is important because without it, the plants that are outplanted on reefs could just as likely be swept away with the tide and out into the open ocean. Outplanting of native species is something that we see done time and time again here in terrestrial systems. On this island, ohia and koa are outplanted in forest reserves by the hundreds to help recuperate forests. Why can't we do the same thing for our undersea forests? During reef recovery projects, cultures of algae can either be outplanted or maybe transplanted onto reefs to help rebound population. Reefs in Hawaii just do not have the algal communities that they once did, for some of the reasons that I talked about earlier. And one of the ways we can help those populations is to physically put the plants back on the reefs ourselves. Particularly after cleanup events or removal of invasive species have been done, there's this gap that's left in the ecosystem. And because invasive algae or invasive species are usually the first ones on the scene, so to say, they're usually able to repopulate the area even if they were just removed from it. But we're able to remove the invasives and then outplant fragments or maybe even entire thalli, which is just a fancy word for the structure of the plant, of these native species. If we're able to outplant those on the reefs, then maybe we'll give them the head start that they need to be able to reclaim the reefs that they once flourished in. This has been done by some groups on Oahu with species that are particularly enticing to our honu, again, the green sea turtle, to make sure that they have something to snack on when they're in the area. If we understand the minimum size that is required for a plant to successfully reattach, or the conditions needed in order to attach in the shortest time frame, then we can ensure that we're creating the largest stock of plants that will be successful when outplanted on the reefs. What this experiment demonstrated is that there's not necessarily a critical size necessary when you're growing these plants for reattachment scenarios, but there may just as well be other factors that impact this likelihood. Size just doesn't seem to be one of them. Now, before I move on to my time at Oceanera, I just want to point out one thing. I designed this experiment based off the work of a previous TCBES student. And the results, I like to think, open the door for future students or even just other future scientists to really get to the bottom of what factors have an impact on reattachment potential. So now, let's switch gears and see how I spent the other half of my internship time. Oceanera is a Kona-based aquaculture facility that not only grows finfish, but also native Hawaiian algae. And with a grant through the Department of Energy, is exploring the potential of these species to be grown at a large scale in a novel offshore array. Formerly named Kampachi Farms, Oceanera created the state-of-the-art offshore pen off of the Kona coast as part of the Valela project, which is pictured here on the right. This was really the first of its kind. It was an unanchored net pen for the aquaculture of finfish, and back in 2012 was named one of the best inventions of the year. They've continued projects focused on offshore production since then, and as previously mentioned, are funded through the Department of Energy to explore the feasibility of an offshore array for growing native Hawaiian algae. The prototype for that design can be seen here on the left. But before algae can be put on such a large array like this, a lot of information about growth rates, reproductive events, even just durability in these open ocean conditions has to be explored. And this is where my work at Oceanera fits into the organization's larger picture. I spent my time here with Ms. Keely Martin, the LIMU technician on the project, who herself is a marine science, Hilo marine science grad. A significant portion of my time in Kona was spent learning the ins and outs of an aquaculture facility. A lot of time was spent cleaning and calibrating the tanks to make sure that the algae are getting the proper conditions needed in terms of cleanliness, water flow, light, and space. The two photos on the left of this slide here are what the before and after of a tank cleaning would look like. You can see in the furthest left photo is a Halimania tank, which started out looking less than ideal. There's a ring of other types of algae and diatoms growing just under what is the standing water line when the tank is full. But with a sponge and a little bit of elbow grease, I got the tank looking better in no time, as you can see here in the middle photo. In addition to keeping tanks clean and keeping these algae happy, I was also able to be a part of working through some trials of algae reproduction. Here in the right-hand photo, you can see me looking just happy as a clam, painting zygotes of sargassum onto a rock. And you did hear that correctly. I was painting them like with a paintbrush, one that you would just get from Home Depot. So after we've collected these zygotes, or the baby algae, we strain them out using really, really fine sieves till all the water is strained out and we have a really thick paste that is actually best picked up with this paintbrush, and then it can be spread across rocks. Then the rocks are left in a tank or multiple tanks, depending on how many there are, where they're protected from the sun with shade cloths to prevent scorching of these little tiny plants. And incrementally, they're given nutrient-rich deep sea water to facilitate their growth. In my time here at Oceanera, I got to see this entire process, from the attachment of these zygotes onto rocks with the paintbrush, all the way through until those plants grew up and actually grew their own reproductive structures, which was a great experience. The day-to-day of an aquaculture site also includes constructing things that you need with the materials available, as well as repairing those that are around you. Here are a few of the projects that I put together during my time at Oceanera. This bottom right-hand photo here is one of the drainage standpipes, a barrier for that I created for a tank of ulva. Ulva is a really leafy green algae. Think of wilted lettuce, and it's really easily broken apart. This barrier was made in order to keep the algae from coming into contact with a pipe that had a really strong suction from the drainage line. So that barrier was therefore a protection from the plants so that they would not be ripped up. In the top right, you can see that shade cloth that I mentioned in the last slide, which not only did I get to repair a few of, but make a new one during my time at Oceanera. This involves sewing fishing line through the holes of the mesh around this PVC frame to ensure that the plants don't get sunburned. The middle photo here is some air lines that are found in some of the experimental tanks at Oceanera. The black tubing along here is the porous air line, which without support would float to the top of the surface of the water. So I worked on making and attaching these PVC frames so that the air line sits along the bottom of the tank and appropriately aerates the plants. This last photo here on the left shows a stand that I made that would allow the filter bags to dry out completely after they're cleaned. The filter system became a focus of my time at Oceanera, as I'll explain in just a moment, and this stand was my very first introduction to it. This is actually one of the first things that I did on my first day on site at Oceanera. Little did I know how important the filters would become. This photo shows the stand while it's not in use, but once the filter bags are taken out of the system, they can be rinsed, bleached, and set upside down on the stand to ensure that they're able to dry out completely before being used again in the next cycle of cleaning. So the filter system on site at Oceanera became really important as it allowed me to update and improve upon the standard operating procedure, or SOP, for cleaning it. Not only did I learn how to clean the entire system myself, but I created a detailed document with photos to ensure that anyone who came on the site could clean the system should that be necessary. I did this first by writing up the procedures, then walking through and taking photos of each step and creating labels for the valves, creating labels for the pipes to ensure there was a continuity between the instructions and what is the physical on-site plumbing. Creating this document allowed me to contribute to the Oceanera organization for years to come as my time with them came to a close. So the photo here on the left shows the main pump and filter house for the entire algae site at Oceanera. For reference, here's a picture of as much of the algae site as I could get into a single photo. A fun side note is that this photo was taken from the top of the boat that Oceanera has that will hopefully soon be in the water taking these algae offshore. This photo highlights the array of tanks that are found on site. You can see these long blue tanks, which are the most plentiful on site, and they're often used for experiments because with so many, you're able to include appropriate replicates. The larger tanks in the foreground here are used to hold and grow the standing stocks of different species, so that there's always some biomass of them available. In the background, once we begin to get these round tanks, that is what begins and marks the fish site of ocean era. As I mentioned earlier, they're also a finfish aquaculture at this organization. The week this photo was taken, there's not many experiments taking place, which is why so many of the tanks are uncovered, because they don't have algae in them. When there's plants in them, they're covered by shade. Also, before I leave, just look at the location of this site. How could you not love this, right? If I remember correctly, during my entire time at Ocean Era, I only remember it raining one day. So you just can't get much better than that. But back to the focus, back to the filter system. So you can see that there's these two bays on the filters here, which use different sizes of filters to ensure the correct purity of water. During my time cleaning these, I really came firsthand to see how important they were. As one time when we cleaned them out, a snail had entered the filter system and released all of her eggs, which hatched into tiny baby snails. So we took them out and there was dozens, if not hundreds, of these baby snails, similar to those that can be seen here. At the top here is what would be the adult snail, similarly sized to the one we found in the filter bag that day. And then at the bottom is a similar size to one of those baby snails that, along with its brothers and sisters, all hundreds of them, coated the inside of these filter bags. If these had been pumped on site into the tanks of algae, they could have posed a major problem for the stocks of algae on site, particularly detrimental to the experiments conducting, which could be super dependent on set amounts of plants. So even the tiniest snail can eat a lot more algae than you think. The middle photo here shows the inside of the nursery container, where water is entering from the outside after being pumped and then is plumbed into various racks, bays, and tanks. This is especially one of the important places where I had to ensure continuity of labeling in the standard operating procedure I created to ensure that the valves get turned on and off in the correct order. You don't want to overfill any tanks or backlog any of the plumbing. It's a lot larger than it may look in this photo, as the width of the rack is more than doubled, and then it's also mirrored on the wall that is behind me. So it's really important that anyone that reads the SOP is able to understand what valves they should be looking for and where they might find them. The final photo here on the right shows part of the filtration system on the outside of that nursery container. This is just half of the filters that the water must go through before entering the nursery. And that includes running through a UV system, which you can see just a little bit of on the very edge of this photo. All of this filtration allows for the water to be as clean as possible so that when rearing these really young plants and conducting experiments, there's less worry about contamination. And I say less because it's almost impossible to avoid completely, but we use all this to do our best and ensure that these plants have their best chance of survival and really providing Ocean Era with that information needed to hopefully one day soon get offshore. So before I wrap it up here today, it would be impossible to describe my experience as a TCBES graduate student without acknowledging the opportunity that I was given as a graduate teaching assistant in the Marine Science Department. Pictured here, you can see me very diligently. My eyes look closed, but I promise they're not. Very diligently watching over some of my fall 2020 students as they took their final. This final, I was given some reference material, but was able to more or less design on my own. Over the 2 years here, I taught 8 sections of the Marine Science 171L, the Introduction to the Marine Biology Lab. And as Carla mentioned earlier, this semester, I got to co-instruct the 171 lecture with her. This experience has allowed me not only to grow as a student, but as a person and an educator. And I am eternally grateful for the experience that I got to teach in a real university setting. Adjusting my teaching to not only an online setting, but also what's pictured here, this hybrid in-person, ultra sanitized room with gloves, That's undoubtedly prepared me for the future that I really hope to find in education. I highly recommend that future students explore the possibility. It'll give you a lot to do. There's a never-ending pile of assignments to grade, but it'll be one of the most gratifying jobs you could have. When you get to take people snorkeling for your job and you find things like this amazing Spanish dancer that we did, it's really hard not to love what you do. So, to reiterate, I could not be more thankful for the opportunities provided to me throughout this internship experience. Lisa, thank you for your unwavering support of me as well as the rest of us internship students. Carla, thank you for taking me in as your grad student and allowing me to teach alongside you. Keely, thank you for being not only my mentor, but also a great friend. I couldn't have gotten to this point without the rest of my cohort, a few in particular. Thank you, Carm, for always making the drives to Kona super fun. To Andre for carrying my buckets of water when I asked, for listening to me talk about algae for hours on end, and just about everything else that you do. And to Dawn, I think you're here too, for not complaining when I asked you to clean my slimy buckets with me. So to wrap things up, I would like to thank all of you here on the Zoom, as well as those that are there on the live stream, for being here today and listening to my presentation. My contact information is in the bottom left corner. I'm always happy to talk about algae or just about anything else for that matter. And with that, I would be happy to take any questions. Mahalo for listening.
Thank you so much. That was fantastic. So if you have a question, feel free to type it into the chat. We have the time for 1 or 2 questions. Or if you're brave, you can feel free to unmute yourself and go ahead and ask it.
Lisa, do you want me to stop sharing or should I leave this here?
Oh, why don't you leave that up for right now?
Okay.
That'd be great. Awesome. Well, okay, I will, I just want to ask Tracy, she's monitoring the live stream. Tracy, do you have any comments there? Not yet. Okay, awesome. All right. So Jordan, what I'd like to know is out of all your experiences that you had at the TCBES program, what do you think is going to be most informative in your journey that you're going to take forward in your career?
In my personal journey, given that I really do hope to find a future in education, I would say that my time being as a TA will really help build upon that. In the short term, I think having practical aquaculture skills for applying for different jobs and just looking around the possibilities of what I can find here on Hawaii Island. I am super grateful that I can say I know how to do some of those things. Basic plumbing, something I definitely did not know how to do beforehand. So yeah, short term and then long term, just having a start and getting to do some education on my own, say that I've taught X amount of classes, I think is going to be a real step up as I move forward.
Oh, that's fantastic. So we do have a question from, we've got some shout outs in the chat, but we have a question from Armando Garcia Ortega. And the question is, from the algae that you worked with, which one do you think has the best potential for aquaculture in the island?
For aquaculture, so of the ones I worked with in my reattachment, Caulerpa lentilifera, the green one that looks like little sea grapes, it's called sea grapes commonly. That one is commonly aquacultured. So as an easy answer, that's going to be the one because it's already commonly done. But I think of the ones that you don't typically see aquacultured, are the ones that I work with. I would say Pterocladiella capisli, the red one that's highly branched, that one reattached pretty well and just general condition wise seemed to do the best in a confined setting of those buckets. Some of the other plants over time did really start to get degraded because some of them live in the intertidal where they're constantly being splashed with new water or they're every once in a while drying out. So the conditions of being completely submerged, as you would typically find in an aquaculture facility, are better suited for some plants more than others.
Wow, excellent answer. Thank you so much for sharing that. So if we all could just give Jordan some sort of sign, thumbs up or Yahoo for her excellent presentation. We thank you so much. We're going to now go ahead and take 5 minutes, a little break, and we'll come back at 9:30 for our next presenter. All right, so we'll see you all at 9:30. Thank you all.
Thank you all.
Aloha, everyone. Welcome back. Our next presenter will be Sebastian Ma'a. He will be introduced by two of his mentors, Dr. Becky Ostertag and Lisa Mira. Dr. Ostertag is a professor in the Department of Biology and the Associate Program Chair of the Tropical Conservation Biology and Environmental Science Graduate Program, both here at UH Hilo. She is a tropical forest ecologist that has her PhD in botany. And when she's not teaching or helping her students, she is working on a variety of research projects, which includes Liko na Palina, hybrid ecosystem projects to better understand how tropical forests are structured and how tropical ecosystems function. Mrs. Lisa Mira is the head administrator for the County of Hawaii's Department of Finance Real Property Tax Division, where she works diligently to fulfill the county's role and mission of assessing all real property and collecting over $350 million in annual revenue. Which of course makes our island work. Her professional goal is for the public to feel appreciated and understood through humble servant leadership within the public sector workforce, which is accomplished by strong ethical and moral standards in which she exemplifies in her daily life as wife, mother, and head administrator for the Real Property Tax Division. Please join me in welcoming Dr. Ostertag to the mic.
Aloha, it's really my pleasure to introduce Sebastian Ma'a today. I've known him as an undergraduate and then as a graduate, and I've had the pleasure of working with him on a project for the last 2 years. And I say it's really a pleasure because Sebastian is truly one of the most kind and considerate people, and it's just a joy to work with him. He's put in a lot of hard work and spirit into his project, working with the Real Property Tax Office with the County of Hawaii. And so I want to pass off the mic to Lisa Mira, who's worked with him through the agency, and we'll say more details on the context of his work within the county.
Hi there. Can you hear me okay, Becky? Okay.
We can hear you great.
Okay. I'm so very honored and excited to introduce Sebastian Ma'a to all of you this morning. He has done much more than just an internship with the County of Hawaii, and his work has made a tremendous impact as stewards of the program. Restoring our land to native forests is not just time consuming, it can be physically exhausting and financially costly. Sebastian has made a clear path for anyone who doesn't have a vast amount of native forest knowledge to dedicate their property to a program which actually does what it's intended to do. It improves our island in so many ways through native forest restoration. There is a lot of talk, especially in government, about public-private partnerships and what we're doing to move forward with island sustainability. But Sebastian's work has actually put this into play. It's not just a plan that will sit on a shelf or an ideal that will never come to fruition. The county law has changed, and he was a huge part of this. It is with great pleasure that I'm here to introduce Sebastian Ma'a to you, who not only is a fellow Konoaina alumni, I had to throw that in there, but he's sincere and leads by example through his passion and perseverance. I'm looking forward to seeing what amazing long-term effects Sebastian has on our island. But first, let's hear from Sebastian on expanding the capacity for the preservation and restoration of native forest habitats on our beautiful island of Hawaii. Thank you.
Aloha, everyone. Just give me a moment to get my presentation pulled up. Can you folks see that?
Yes.
Yeah? Okay, awesome. Well, thank you very much, Lisa and Becky, for that introduction. That is truly humbling to be able to have worked with you over the past 2 years. And thank you very much for sharing all the knowledge with me that you have. Thank you, Jordan, for that presentation. That's going to be a really hard one to follow, but I'm going to try my best. And thank you to Carmelita and Kavehi for getting us grounded today and for kicking off this symposium. And with that, I'll just hop right into my presentation. So aloha kakahi aka kako. My name is Sebastian Ma'a. And today I'll be presenting my journey as a tropical conservation biology and environmental science professional intern who had the opportunity of working with the County of Hawaii Real Property Tax Division under the guidance of Lisa Mira, Dr. Rebecca Ostertag, and Lisa Canali. I want to thank you all for taking the time to be here this morning, and I hope that you enjoy my presentation, which is entitled Expanding the Capacity for the Preservation and Restoration of Native Forest Habitats on the Island of Hawaii. Now, before we begin, I just wanted to let you know that there'll be a short period of time at the end of my presentation for questions and answers, and that unless there is a photo credit associated with the pictures in my slides, then the pictures are ones that I've taken on the journey that has brought me to where I am today. And with that, we'll just go right into it. So this is a general outline for my presentation. I will begin with a brief introduction where I'll provide you with a little background about myself and the importance of my internship project. I will then move on to talk about the overarching purpose of my internship and the approach that I used to successfully conduct my project. And once that is complete, I will discuss the outcomes of my project, the broader implications of my work, and I will then end with a brief conclusion to tie everything together. So out of all the places that I could have been born, I feel very blessed to say that I was born and raised on the Big Island of Hawaii. Now, although my time living on this island paradise has been relatively short, I have watched my beloved childhood playground change dramatically right before my very eyes. I come from a very humble background where I grew up living on a coffee farm, climbing trees, learning how to fish with my dad and two younger sisters, and spent many days surfing from dawn until the sun disappeared, forcing me out of the water. These experiences is why I say that Hawaii is my playground, because growing up the way that I did, the best things in life were free, as there is no price tag when it came to getting outside and enjoying the natural beauty that surrounds us. Prior to starting the academic journey that has brought me to where I am now, I spent 5 years as the partial owner and operator of a successful landscaping business that specialized in maintaining high-end residential and commercial properties in the District of North Kona on the island of Hawaii. As time passed by, I reached a point where I could no longer contribute to the unsustainable use of our shared natural resources, the rapid transformation of the beautiful playground that I like to call my home, while also struggling with the fact that my efforts were only serving a very small fraction of the community here on the Big Island. So in 2016, I decided to sell my business and enrolled at Hilo as an undergraduate in the Geography and Environmental Studies program, which unknowingly started the journey that has brought me to where I am now today. So following my first semester as an undergraduate in the fall of 2016, I had the opportunity of participating in the Pacific Internship Program for Exploring Science, also known as the PIPES program, for 2 consecutive summers, where I did water quality monitoring at Kaloko Fish Pond and local i'a rehabilitation at Aimakapā Fish Pond at Kaloko Honokohau National Historical Park in 2017. And then in 2018, I assisted with bioacoustic monitoring, arthropod surveys, and developed a Mamane plant monitoring protocol for the Office of Mauna Kea Management. These experiences not only solidified my passion for preserving the place that I call home, but more importantly, PIPES reaffirmed my career aspirations of working in the field of conservation. It also helped me see firsthand by working and learning from tropical conservation biology and environmental science graduate students like Brett Mossman and previous TCBES graduates like Jessica Kirkpatrick and other natural resource management professionals, how the activities that take place at Mauka have a direct impact on the health and productivity of coastal ecosystems, which helped me understand the importance of sustainable ridge to reef management. So although I don't have an extensive background in conservation related work, and I didn't spend a lot of time working in and learning about native ecosystems growing up, the experiences that I had as an undergraduate really reminded me that while it is a blessing to be able to say that I was born and raised on the island of Hawaii, that blessing also comes with a deepened sense of responsibility or kuleana to preserve and restore the natural resources of Hawaii for present and future generations. It is that responsibility that ultimately motivated me to take my education to the next level, and I saw the TCBES program as a way to make that happen. Now, during my time as a TCBES graduate student, I was able to take the vegetation of the Hawaiian Islands class, where I learned about plant identification and the wonders of native forests. So what started as a class eventually turned into one of my new favorite hobbies, which is spending time in the forest and studying native plants. Now, while I don't consider myself to be a botanist, I have developed a deep admiration for native forests and have come to appreciate them in ways that I never did before. So the things that I learned in that class, in addition to the concepts of habitat degradation, the loss of biodiversity, invasive species, and being able to venture out and see these conservation-based themes right before my very eyes, really motivated me to do what I can to preserve and restore our fragile native forest ecosystems. So in the fall of 2019, during my first semester as a graduate student, when I was approached by Dr. Rebecca Ostertag and was presented with the opportunity of assisting with the development and implementation of new legislation that was geared towards increasing the capacity for the preservation and restoration of native forest habitats on the island of Hawaii, I felt like I had found a way to make my dreams of preserving native forests and also serve the community of Hawaii Island become a reality. So Becky pulled me under her wing, where I was able to join the Native Forest Dedication Program working group, which consisted of an environmental lawyer, Leslie Cole Brooks, the division head administrator, Lisa Mira, and Kita Joe, who is the assistant administrator of the County of Hawaii Real Property Tax Division, and Dr. Susan Cordell, who is the director of the Institute of Pacific Islands Forestry, to help them expand the County of Hawaii Native Forest Dedication Program. So my role as an intern with the Hawaii County Real Property Tax Division was to develop supplementary materials that will support the expansion of the Native Forest Dedication Program. Much like the llama and the iliahi or sandalwood tree pictured here, this included collaborating with different staff members at the Real Property Tax Division office, environmental lawyers, ecological professionals, and generating the complementary documents that are necessary for effectively integrating the proposed property tax amendment into the existing County of Hawaii Real Property Tax Code. So the purpose of my internship project was centered around three main ideas, which are: one, advocating for Native Forest Bill 178, two, creating supplementary materials to support the expansion of the Native Forest Dedication Program, and three, participating in public outreach to raise awareness about this newly updated program. So in an effort to illustrate the importance of this legislation, I conducted a comprehensive literature review using Google Scholar, the Hilo online library system, and other peer-reviewed repositories to quantify the economic value of native forests. The information collected during this part of the project was compiled into an annotated bibliography and was intended to provide Hawaii County Council members with a deeper appreciation for the innumerable goods and ecosystem services provided by native forests, and how the County of Hawaii can benefit from the expansion of the Native Forest Dedication Program. So as you can see here from this excerpt taken from a publication by the Nature Conservancy, native forests are a significant economic asset, as a single Hawaiian watershed during the time of this publication in 2010 was valued at $7.4 to $14 billion. The next component of advocating for Native Forest Bill 178 was submitting 3 rounds of verbal and written testimony during the public hearing process of this new legislation. Each testimony was modified to support the passing of Native Forest Bill 178 and was presented at each public hearing, which took place on July 7th, July 22nd, and August 5th of 2020. Written testimony was reviewed by the Native Forest Dedication Program Working Group and submitted to Hawaii County Council before each meeting, and verbal testimony was articulated in person at the Council of the County of Hawaii at 25 Aupuni Street in Hilo. The second component of my internship project consisted of developing supplementary materials to support the newly updated Native Forest Dedication Program. So in an effort to take the guesswork out of helping landowners determine what types of plants they can use to meet the requirements for the different native forest designations, I developed a native, non-native, non-invasive plant species list that helps individuals determine what types of plants they can incorporate into their native forest restoration endeavors. This list was created by referencing different plant resources and guides, like the Manual of the Flowering Plants of Hawaii, growing Hawaii's native plants and plants for tropical landscapes to select plant species that are the most appropriate for the ecological zones found on the island of Hawaii. Plants in this list were selected by growth forms, those that are easily obtained from nurseries, plants that did not contribute to the decline of other native flora and fauna, and those that are relatively common in existing native forest, functional forest, and successional forest environments. Selected non-native, non-invasive plant species were those that had a low-risk score or a score of 0 or less by the Hawaii Pacific Weed Risk Assessment, which is a plant screening tool that botanists use to evaluate whether or not plants are likely to be or become invasive in Hawaii and other tropical Pacific islands that have similar climate regimes. A plant nursery list, which is an accompanying document to the native, non-native, non-invasive plant species list, was created to provide landowners with a directory of where some of the plants featured in the native plant species list can be purchased. Now, once the plant nursery list was compiled, I conducted site visits and follow-up phone calls to ensure that the businesses in this list carried some of the plants in the plant species list, were still in operation, and agreed to be included in this resource. Now, some, but not all of the nurseries in this document are endorsed by the Big Island Invasive Species Committee, Plant Pono website, and Dr. J.B. Friday of the University of Hawaii at Manoa, College of Tropical Agriculture and Human Resources Cooperative Extension Service. Another document that was created to support the Native Forest Dedication Program is the Native Forest Dedication Program Management Plan and Guidelines document, which is intended to provide landowners with the resource they can use to help them navigate the process of qualifying for a native forest land use dedication. With the guidance and support of Tanya Rubenstein from the State of Hawaii Forest Stewardship Program and Matthew Cocking from the Natural Resources Conservation Service, I developed a document that would allow landowners to write an ecologically sound native forest management plan without the help of a natural resource management professional, while also helping them meet the eligibility requirements listed in Chapter 19, Article 8, Section 19-59 of the Hawaii County Code. And the last document that I created was a management plan evaluation checklist, which was specifically designed to complement the management plan template document, making the evaluation of each management plan as seamless as possible. The management plan evaluation checklist document is broken into 3 main sections, with each section including elements that are specific to one of the 3 main native forest dedication types. The list of questions included in each section was written to follow the order of the sections listed in the management plan template and was designed to make sure that the management plan proposals are fulfilling the eligibility requirements listed in the Hawaii County Code. For the third component of my internship, the public outreach component, I helped develop, design, and co-host a Native Forest Dedication Program webinar, which was intended to unpack the new Hawaii County legislation on tax incentives for native forest dedications. The webinar and the information presented at the webinar was developed over a 3-month time period where the Native Forest Dedication Program Working Group had monthly meetings via Zoom to discuss the content that was to be delivered, the format of the presentation, and how to effectively convey this new legislation to the general public. So I am happy to report that although I represent only a very small portion of the individuals who champion this new legislation, that the advocacy of Native Forest Bill 178 paid off, as Native Forest Bill 178 was passed and signed into law on August 17th of 2020. There is overwhelming support for this new legislation, with all 9 Hawaii County Council members saying yes to this new bill, which created 3 new native forest categories. As a result, private landowners of Hawaii County can now dedicate their land to one of 3 native forest land use designations, which includes a native forest, a functional forest, and a successional forest dedication. So as I previously mentioned, Native Forest Bill 178 was passed on August 17th of 2020 and went into effect on January 1st of this year. This new legislation expanded the Native Forest Dedication Program to include 2 types of dedications. A preservation-based dedication, which is designed to maintain current conditions, which specifically pertains to forests that already have native species coverage, with at least 60% native species cover, and a restoration dedication, which specifically pertains to forests that already have, or which is based on improving current conditions to develop a native dominated forest in the future. Also new in Native Forest Bill 178 is the addition of two new categories, which include a functional forest and a successional forest land use designation. So the basic eligibility requirement for each native forest dedication type is that the land parcel be at least 3 acres or larger, with at least 2.75 acres being intact and contiguous native forest. So the first dedication type is a native forest dedication, which Leslie likes to refer to as the gold standard of the program. Under this land use designation, private landowners can dedicate land to native forest preservation or restoration for a 20-year time period, with native forests being defined as an area of land which has 60% or greater native species coverage in the canopy, the understory, or a combination of the two, with land being assessed at a $7 per acre value. The functional forest dedication also has a preservation or restoration option, which also requires landowners to dedicate land for a 20-year dedication period. And for the purpose of this land use category, functional forests are those that have 60% or greater native and non-native, non-invasive plant species cover in the canopy, the understory, or a combination of the two, with the dedicated land being assessed at a $14 per acre value. Now, in this case, as I mentioned earlier, non-native, non-invasive species are those that have either a low-risk designation or a Hawaii Pacific weed risk assessment score of 0 or less, meaning that they are unlikely to be or become invasive here in Hawaii. Now, the last category is a successional forest dedication, which pertains to lands that have new lava substrates in which the soil depth or organic material are less than 10 centimeters and is maintained to promote native or functional forest development over the 50-year dedication period and is assessed at a $100 flat rate. So here are a few examples of the potential savings in property taxes when choosing to dedicate land to the Native Forest Dedication Program. It is important to note that tax savings will vary as the amount of potential savings is dependent on the market value and the size of the dedicated property. In the first example, it is a property located in South Kona that is 6 acres in size. The respective taxes to be paid annually based on the native forest value of $42 would be $200, which is the minimum tax in Hawaii County. So based on the market value and the size of the property used in this example, the annual savings by dedicating land to native forest for this property would be $1,754. The second example is a property located in Hamakua that is 602 acres in size. The respective taxes to be paid annually would also be $200, which again is the minimum tax in Hawaii County. So the annual savings for the property used in this example would be $32,571. And the last example on this slide is a property located in South Kohala that is 22 acres in size. Successional forests, as I mentioned earlier, are assessed at a flat $100 value, regardless of the size of the property. So the respective taxes to be paid annually based on the successional forest land use dedication in this example would also be the minimum tax of $200, with the annual savings being $13,722. So upon completion, the plant species list included 68 native and 66 carefully selected non-native, non-invasive plant species with descriptions of each plant, their preferred growing conditions, and how to successfully cultivate each species. This list allows landowners to correctly identify, select, and cultivate different plant species based on elevation gradients, moisture regimes, and their preferred growing conditions. Now, once this list was compiled, I created another document or another section, which included a photograph and description of each plant species, its preferred growing conditions, and helpful tips on how to ensure plant growth and plant viability. So as I mentioned earlier, I also created a plant nursery list, which serves as an accompanying document to the plant species list, as it provides landowners with a directory of where some of the plants featured in the native plant species list can be purchased. The native plant nursery list includes an alphabetical list of Big Island nurseries that specialize in selling and distributing native plants, as well as each nursery's contact information, including the address of the business, their phone number, and their email address if it was available. And for the management plan template document, I created this table, which was designed to simplify the process of helping landowners select a native forest dedication type that most closely corresponds to their current situation, which ultimately helps landowners decide which land use designation is best for them. So each dedication type listed has a corresponding color that highlights the eligibility requirements for each native forest type, the eligible land use classifications to qualify for the native forest dedication program, and how they differ in terms of native forest structure. Another document that was created to support the implementation of the newly updated native forest dedication program is the Native Forest Dedication Program Management Plan template document, which is intended to provide landowners with the resource they can use to help them navigate the process of qualifying for one of the native forest dedications mentioned earlier. This document includes a simple step-by-step guide on how to dedicate land to one of the 3 native forest dedications, and a management plan template that takes the guesswork out of eligible landowners developing their very own native forest management plan without the help or guidance of a natural resource management professional. Now, the last document that I created was a management plan evaluation checklist, which is intended to complement the management plan template, making the evaluation of each management plan as seamless as possible, while also adhering to the requirements listed to the newly updated Native Forest Dedication Program tax amendment. Now, the management plan evaluation checklist document is broken into 3 main sections, with each section including elements that are specific to one of the native forest dedication types. The list of questions included in each section follows the order of the sections listed in the management plan template and was derived from the eligibility requirements section to make sure that management plan proposals are fulfilling the requirements to be eligible for each native forest dedication. So at the end of the evaluation process, the individual evaluating the management plan proposal should have sufficient information to determine its status and what steps need to be taken in order to proceed. So for the public outreach component of my internship, I co-hosted the Hawaii County Legislation on Tax Incentives for Native Forest Dedications webinar with the Native Forest Dedication Program Working Group on February 11th of 2021, with 48 individuals from the general public being in attendance. This webinar was recorded and later converted into a YouTube video, which is now available for the general public to view at their convenience. So this project has numerous ecologic, cultural, and economic benefits as it facilitates the multi-stakeholder participation in the active care and management of native forest habitats throughout the county of Hawaii. In a time when our beloved ohia forests are threatened by destructive pathogens such as rapid ohia death, planting more native and non-native, non-invasive plant species not only increases the potential for rainwater capture and groundwater recharge and retention, it also increases the rates of carbon sequestration, which may reduce elevated greenhouse gas levels and mitigate the impacts of anthropogenic climate change. From a cultural perspective, as many foundational components of Hawaiian identity are inherently tied to native forest habitats, the preservation of Hawaiian traditions is highly dependent on the health of native forests. So this legislation is one way to ensure that the perpetuation of customary practices that have been handed down throughout the ages of Hawaiian history will be able to continue as they have since they were first developed thousands of years ago. And lastly, from an economic standpoint, the resources that I created has the potential to save landowners and the community members of Hawaii Island substantial amounts of money in both short-term and long-term timeframes, as there is now a standardized protocol that allows the county to streamline the process of evaluating incoming management plans. So the expansion of the County of Hawaii Native Forest Dedication Program to include a functional forest and a successional forest dedication illustrates the efficacy of increased collaboration between government agencies, researchers, and legal fellows to effectively preserve and restore native forest habitats on the island of Hawaii. As of right now, there are 155 parcels participating in the Native Forest Dedication Program, which represents roughly 70,263 acres of land being dedicated to native forest preservation or restoration. Now, while the number of new applicants has been relatively low since Native Forest Bill 178 went into effect earlier this year, the staff at the Real Property Tax Division has reported that there has been an increased interest in this program as they've been receiving more calls about this newly updated land use designation. So my hope is, as the number of applicants increases, that the documents that I created will help the Native Forest Dedication Program bloom and eliminate the barriers that would prevent this program from being successful, as the County of Hawaii Real Property Tax Division and eligible landowners will have the resources they need to successfully implement this newly updated program. The Native Forest Dedication Program is also a model that can be adopted and applied by other counties here in the state of Hawaii, other states around the nation, and other nations around the world. This new legislation, which is the first of its kind in the state of Hawaii, represents the beginning of an era in which community members and the natural environment benefit from the co-production of initiatives created by ecological researchers and political institutions, which empowers the general public to play an active role in the preservation and restoration of native forest habitats. As native forests are primarily managed by various state, federal, and nonprofit organizations, this legislation promotes community engagement in the preservation and restoration of native forest habitats, which may inhibit landscape level degradation from the mountains to the sea. Now, as many novel ecosystems have passed an ecological threshold, making them unresponsive to traditional management techniques, the Global Environmental Stewardship Model and the Native Forest Dedication Program showcases the importance of increased collaboration between various entities to preserve the functional characteristics of traditional and modern ecosystems, to help them become more resilient to natural and anthropogenic landscape level alterations now and in the future. So in conclusion, I just wanted to end my presentation with a picture of this lama tree, which is associated with light and enlightenment in the Hawaiian culture. While it feels like it was only yesterday that I first started the graduate school journey that has brought me to where I am now, when I look back at my experiences during the past 2 years, I'm truly humbled to say that I have found a way to use the skills and knowledge that I've gained by working with the Native Forest Dedication Program Working Group to implement legislation that allows the community members of Hawaii Island to work towards perpetuating the undying legacy of our native forest habitats. Through the various projects that I have worked on, I can now say that I have the ability to navigate the process of developing government level documents without constant supervision. And through the process of conducting nearly this entire internship remotely, it also taught me how to coordinate and plan meetings with team members, how to effectively communicate project setbacks and progress to the Native Forest Dedication Program Working Group, and how to make positive contributions to a team through my actions and personal undertakings. Now, one consistent theme that appeared regardless of what I was doing was the importance of collaborating and working diligently to achieve group and organizational goals. As the world is faced with many seemingly insurmountable challenges in the form of climate change, the loss of biodiversity, and large-scale habitat degradation, my experiences over the past 2 years reminded me that no matter how small my efforts may be, that I too can play an active role in resolving and working through these and many other issues to make this world a better and brighter place for those who are yet to come. So while at times it may feel like the things that I do are insignificant in relation to the many adversities that the world is currently faced with, when I reflect on the lessons that I have learned during my educational journey in the TCBES program, I'm reminded that nothing is impossible when people unite for a common cause. So here's a list of some additional resources. At the top, you can find the County of Hawaii Real Property Tax Division website, where you can find the link to the management plan and guidelines document, the native, non-native, non-invasive plant species list, and the plant nursery list. And on the bottom is a link to the Native Forest Dedication Program webinar. And lastly, before I finish, I'd just like to send a very sincere and heartfelt mahalo nui loa to Becky for the unwavering support you've provided me throughout my journey through graduate school. To Lisa Canali for all the knowledge you've instilled in me as a TCBES professional intern. I would like to thank the Hau'oli Maoloa Foundation for your very gracious and generous funding. And I would also like to thank the Native Forest Dedication Program Working Group, Leslie Cole Brooks, Susan Cordell, Lisa Mira, and Kita Joel for all the knowledge you've shared with me while working on this project and for everything you do to preserve and protect our native forest habitats. I I would also like to thank the PIPES program and program leaders, Sharon Ziegler-Chong, Linnea Hugh, Rita Miller, and Jordan DeJesus, for showing me a path towards a career in conservation and helping me grow into the individual that I am today. And lastly, I would just like to thank my family, my friends, and my cohort for always being there for me during this crazy journey through graduate school. There's no way I could have done it without you guys, so mahalo. And with that, I just want to end with this mahalo slide. At the bottom, you can see my email address. So if I'm not able to answer any questions during our question and answer session, please feel free to shoot me an email and I'll happily do what I can to get those answered. And with that, I want to thank you all again for being here to listen to my presentation, and I will now take any questions. Mahalo.
Thank you so much, Sebastian. That was fantastic. If you have a question, please feel free to put it into the chat, or if you'd like to be brave, please feel free to unmute yourself. And there's also a comment area on the live Zoom, so you could put a question in there. So Sebastian, while we're waiting for a question, I have one for you. You had an opportunity to do advocacy work at the county level, and I'm wondering what sort of advice you would give a person that's new to that to be able to give effective testimony and to advocate for their causes.
Okay, yeah, that's a great question, Lisa. Thank you for asking that. This was my first time ever participating in any type of public outreach or public testimony, so I was a little bit unsure about the process, but you know, the people that I worked with, Leslie Cole-Brooks, Becky, and the rest of the staff at the county, they really helped prepare me. So I think the biggest thing that I would recommend is just doing your homework and familiarizing yourself with a background of what you're trying to advocate for or against, and then also designing the things that you want to say to help deliver that message in an effective way. I also think it's really important to think about your motivation for participating in public outreach, you know, and how you fit into that equation. And I think that really goes a long way in helping to develop and articulate effective verbal and written testimony during the public hearing process.
All right, thank you so much. So Carla, go ahead and take it away.
Just a real quick, it was fabulous. And can you go back to the slide that has all your websites so I can copy some of them down?
Yeah, hold on, no problem. Here you go.
Thank you. Thank you. Okay, keep going.
Does anybody else have any questions?
So Tracy, how does it look on the live stream?
Nobody's using the live stream. They're watching it. They're watching it. They're not typing in there. But I did have a question, and I remember from the symposium too. Why is the native forest less per acre than the functional forest?
That is a really good question, Tracy. To be honest, that is something that I'm still trying to grasp my head around. You know, I do not have a lot of experience when it comes to the development of the different pay scales of how these different native forest dedications are evaluated in terms of the amount of property tax breaks that people receive. But that is something that I could look into and get back to you if you would like.
Here we go. Hey, can you hear me? Sorry, it's a little noisy up here. Sebastian, that was excellent. Your work, you know, I don't know if people realize how incredibly valuable it is, because what happens is laws get passed and then they just get dusty somewhere and people don't use them because they don't know about it or they don't know how to start. And you know, you just dug in, and all of those documents that you created to help people first inform them what they need to do and then guide them along the way. It's just so important. I was really reminded and impressed to see all the documents up at how much work you've been doing. And you know, people don't realize, like, now it's going to go from an idea and a concept to a real life thing. And I can't wait to hear about the applications rolling in. So I have two questions for you. First, I'd like to know, what if you could describe your ideal job now that you've had a little taste of government and legislation and advocacy and you're out in the field and you know, if you could just imagine your ideal job, I'd like to hear about what that's going to be next. And also I was curious on a more practical, just with the docs that you did, how can someone discover what their what biome they live in? You know, what's the plant list like? Where's it best to start if you're not really knowledgeable about plants, but you know that some would work and some won't? Is there a place in your documents that kind of helps direct someone? So, so those two questions. I'd love to hear what you had to say.
Awesome. Thank you, Leslie. Those are really good questions. So as of right now, I currently do not have any job positions lined up after school. But I would really like to take this project a step further and use my the knowledge that I've gained over the past 2 years, plus the things that I have in my background of landscaping, to help people implement this legislation and develop their very own native forest management plans, do site evaluations and visits to help people get on track for doing a native forest or a functional forest or successional forest dedication. I don't know exactly where I would fit into that equation or what organization I would be working with, but that would be something that I would really like to do, is just to get my feet on the ground and start putting this in, this legislation and the things that I developed into action. And then in terms of helping people determine what their ecological zone is, there's actually some resources in both the management plan guidelines document and the native plant nursery, or the native plant species list, where there's a directory that'll help people determine their elevation gradient, where they're located on the slopes of the island, and then also their moisture regime. So which type of what's their rainfall gradient and where they would fall into that. So those are two really big determining factors. And then there's also Cheryl Chan from the County of Hawaii Real Property Tax Division created a couple of maps that the general public can use to help them locate, or a general area of where they're located and the types of moisture regimes and elevation gradients that they are in. So I think those are two resources that the public can use in helping them determine where they fall in the ecological spectrum of the many ecosystems we have here.
Awesome. Thank you, Sebastian. That was a very thorough answer and a fantastic presentation. And so just give yourself a big pat on the back, because that was marvelous. What I'd like to do now is we're going to start our next presentation in 2 minutes. So let's just all just take some breaths, maybe look away from the computer for a couple minutes, and then we'll come back at 10:15 for our next presenter, Sarah Norbaum. Thank you all. We'll see you in a couple minutes. All right, welcome back, everyone. I am very excited to introduce our next presenter, Sarah Norbaum. She will be introduced by her mentor, Tamara Entz. Tamara was most recently the Habitat Assessment and Monitoring Coordinator at a nonprofit in the North Coast of Oregon called Tillamook Estuaries Partnerships. I'm so sorry, it's Tamara. Tamara's path led her through the eastern grasslands, black spruce bogs, and the sagebrush desert, where she has conducted several plant and bird species and bird surveys, and also the Arctic islands, where she performed a whale census, which then finally washed her downstream into a Western estuary and the role of Habitat Assessment and Monitoring Coordinator, and Sarah's mentor. I had the great pleasure of meeting with the two of them every month, and it was super fun. I'm going to be missing our monthly meetings. And so please join me in welcoming Tamara to the floor.
Hello, everybody. Welcome. I'm glad to see you all here and happy to introduce Sarah. Sarah came to me through Tillamook Estuaries Partnership looking for an internship. And given COVID at the time, we were not actually offering internships. So I pulled a few strings and I'm eternally glad that I did. And Sarah is at her best in nature. Her core and future are learning about and protecting the environment. She has the lofty goal of making a difference, and she wants to know in the end that she tried. And I think she has more than tried and has performed beautifully and admirably, and her legacy at TEP will be synonymous with mine. So I'm very grateful for her and all the work she did. And with that, I will turn it over to Sarah.
Well, thank you for the introduction. And also thank you to Sebastian and Jordan for your guys' wonderful presentations and all the hard work that you guys have done. Let me get this started. Is everyone able to see that okay? Thumbs up? Okay. So aloha, my name is Sarah Norbaum, and I'd like to thank you for coming to my presentation on my professional internship project that I completed as part of my graduate requirements for the Tropical Conservation Biology and Environmental Science program at the University of Hawaii at Hilo. My presentation is titled Surveying the Oregon Coast, Work in Conservation and Preservation of Estuarine Systems. As a note for this presentation, any pictures, figures, and maps without credits are my own creation. For this presentation, I'll give some brief generalized information about estuaries, discuss my internship organization by answering the questions of who they are, what are their mission statement, where are they located, what do they do. I will then discuss one of the estuaries I spent a lot of time at, which was located at Sitka Sedge State Natural Area, and discuss the water quality monitoring we were doing at this location. Then I'll move on to discuss the Seeds of Success program and my involvement with seed collection, then discuss the larger part of my internship project, which was GIS data collection and compiling it to make maps, and then what I did for advocacy and learning the agency ecosystem, despite COVID restrictions. And then we'll finish it up with a conclusion and acknowledgements. To first define estuaries, they are partially enclosed areas that appear to be a transition from land to sea. Estuaries are influenced by the tides and are places where fresh water from things like rivers and streams would mix with salt water. Estuaries go by many names and have been called a bay, a slough, a lagoon, or a sound. They also consist of many different habitat types, such as any combination of like a shallow, open waters, fresh and or saltwater marshes, swamps. They could be more sandy beaches, mud flats, sand flats, rocky shores. They could have reefs, oyster reefs, mangrove forests, deltas, tide pools, and seagrass beds. Estuaries are unique because they are all different from one another. Estuaries are an important biome that are considered to be one of the most productive systems on Earth due to the ecological and economic goods and services they provide. Examples include protection of coastal communities by preventing erosion, stabilizing shorelines, and protecting communities from harmful effects caused by extreme weather events such as flooding or hurricanes. Estuaries act as a natural buffer to absorb flooding waters and dissipate storm surges. This is due to the plants and soils surrounding the estuaries acting as a sponge that soak up floodwaters, and the sand dunes and marshes surrounding the estuary depleting the strength of waves during storm surges. They also act as a filter to minimize inflowing pollutants and excessive nutrients before water enters the ocean. They support a diverse community of plants and animals by providing a place for these species to live, reproduce, nest, and feed. In terms of the economy, estuaries provide approximately $800 billion each year in trade and commerce for the United States and generate an additional $8 to $12 billion in tourist revenue yearly. Overall, 28 million jobs in the United States rely on a healthy estuary ecosystem. Despite estuaries contributing so much and playing a critical role in human development, estuaries and other coastal ecosystems have been declining at a significantly accelerated rate due to multiple confounding impacts such as a loss of species, pollution, increased nutrients, decreased water quality, increased invasive species, and overall loss of area due to human activity. This is why organizations like my mentor organization are needed to address multiple issues that affect estuary systems and come up with management plans to conserve, restore, and/or protect estuarine systems. That is why I wanted to work with my mentor organization, Tillamook Estuary Partnership. I was fortunate Tamara Enns, who worked for Tillamook Estuary Partnership, was willing to take me on as a professional intern and come up with this wonderful project that would give me so many experiences. Tillamook Estuary Partnership is a nonprofit organization with the mission to conserve and restore Tillamook County's watersheds through active stewardship, scientific inquiry, community engagement, and education. Tillamook Estuary Partnership is located on the north coast of Oregon in Tillamook County. The Office for Tillamook Estuary Partnership, which I will now refer to as TEP for the rest of the presentation, is located in the small city of Garibaldi, and it's on the longest pier in Oregon, which overlooks Tillamook Bay. However, TEP works beyond their offices. You can see on this map the study areas TEP focuses on for their work. Each region area on the map was designed by the rivers and watersheds they encompass. TEP is one of the organizations involved with the Environmental Protection Agencies, otherwise known as EPA, designated 28 national estuary programs. This program designates estuaries of national significance on the Atlantic, Gulf, and Pacific coasts. The EPA provides funding, guidance, and technical assistance to the local national estuary programs to develop and implement comprehensive conservation and management plans. These documents are long-term plans to address the challenges and priorities of conserving, restoring, and managing the estuaries and the surrounding watersheds. Tillamook Bay was designated as a Bay of National Significance in 1994, and since then TEP has not only worked to protect and restore the health of Tillamook Bay, but all the estuaries and watersheds located in Tillamook County. Since I was interested in learning more about working in conservation, restoration, and management of estuaries and coastal systems, TEP was the perfect organization for me to work with. And one of my first projects with them involved gathering data for an estuary that they recently agreed to help monitor and, if necessary, restore. So our job was to get like an initial baseline of the water quality at this estuary. So Sitka Sedge is located south of Tillamook and is part of the Sand Lake Estuary. This area includes about 357 areas of mudflats, dunes, wetlands, and tidal marsh. The Sitka Sedge area was diked and farmed during the 1900s, and since 1989, many residents in surrounding communities have worked to preserve the site. However, in the early 2000s, the property went on the market and golf course developers were attempting to buy the property. They had made several proposals for the golf course, but the community members opposed the proposals. And the Terra Del Mar community in particular had worked to prevent the area from becoming developed. In 2014, EcoTrust purchased the property, and later that year, Oregon Parks and Recreation Department acquired the area from EcoTrust using both the Oregon Lottery funds and a National Coastal Wetlands Conservation Grant. In June 2018, Sitka Sedge State Natural Area was open to the public. So why do we need to monitor at Sitka Sedge State Natural Area? Well, in the 1930s, the area was used as a dairy farm, and a dike and tide gate were built to provide a pasture for cattle and a road for vehicles. The first gate had failed and was replaced in the 70s, but that tide gate has deteriorated since that time. The main problem with the dike and the tide gate, however, is the location, and the way the dike is built limits the tidal exchange within the estuary and impedes the movement of native fish such as the salmon and trout. Oregon Parks and Recreation Department has been trying to address the issues and have been collecting data on the area to discuss the issues with the dike and come up with an alternative plan that would improve the area for a fish passage while also not increasing the chances of flooding in the surrounding communities. The Oregon Parks and Recreation Department held a public meeting in January 2020 to discuss habitat restoration designs and the ideas for what can be done with the dike. But after the meeting, COVID-19 became an issue, and that led to budget cuts within the Oregon Parks and Recreation Department. Due to the budget cuts and loss of staff, the department had issues with being able to collect additional data needed at Sitka Sedge State Natural Area. So TEP and other agencies have stepped in to assist. Some of the data needed was information on current water quality near the dike and tide gate at Sitka Sedge State Natural Area. We needed to know the basics such as the average water temperatures and dissolved oxygen levels on both sides of the tide gate. So the equipment we used for this data collection project were 4 dissolved oxygen loggers, which I'll now refer to as DO loggers. These loggers were calibrated and set to collect data every 15 minutes. These DO loggers were able to collect the dissolved oxygen of the water along with the temperature. Then we used PVC pipes as a protecting casing for it, for the equipment, and then zip tied them to T-posts we placed in the water at low tide. So this map shows the placement of the 4 DO loggers, along with the names we used for the loggers, so we were able to keep track of the data, which data belonged to what logger. And as you can see on the map, Sand Lake 2 was placed on the north side of the tide gate, and Sand Lake 3 was placed on the other side, with the 2 loggers being placed further south towards the river that fed into the estuary. Sand Lake 1 and Sand Lake 2 were the first loggers we placed. Then approximately a week and a half later, we went back out and placed Sand Lake 3 and Sand Lake 4, since we had the equipment and wanted to get additional data. So once the loggers were placed, we used an instant read DO probe and placed it next to the long-term logger for a comparison baseline. The DO probe would tell us the temperature, the percent of dissolved oxygen, the level of dissolved oxygen, and the barometric pressure. Then we would return 2 weeks later to check to make sure the loggers were in place and to drop the DO probe again for that comparison. Then 2 to 4 weeks after the long-term dissolved oxygen loggers were placed, we went back and pulled the loggers from the water, and I took them to download the data using Hoverware software. The data was then converted into Excel sheets where I was able to clean up the data and complete a simple data analysis. So I calculate the mean, median, mode, and range of the dissolved oxygen levels and temperatures for each logger and create a box plot to show the comparison of the 4 sites. In addition to this, there were charts created for each location that included the tidal data and precipitation rates. So the mean dissolved oxygen levels should remain near 5.5 milligrams per liter for optimum growth and survival for many fish species. But they could go down to 2 to 3 milligrams per liter for brief periods of time. So the relieving part of our data was that the mean was able to stay near the optimum level for most of the sites except for Sand Lake 1, which had a mean of 5.14 milligrams per liter. However, all 4 sites had periods of time where they were either at the minimum range the DO could be for brief periods of time or below it, with the lowest DO reading being at 0. Overall, it showed how we needed to gather more data on this area. And due to the monitoring being pushed into the late fall, it also showed how it would be beneficial to continue to gather data, not just in the spring and summer months, but also in the fall. So the next project that I briefly assisted in was for the Seeds of Success program. The Seeds of Success program is led by the Bureau of Land Management and was established in 2001. The Bureau of Land Management partners with both federal and non-federal agencies and organizations to collect native seeds. These seeds are used for research, development, and conservation and restoration. The Northwest Oregon Regional Partnership is a program that is part of TEP and one of the organizations that is part of the Seeds of Success program. Our goal was to collect over thousands of seeds from multiple parent plants so that they could be sent to be processed, and then any leftover seeds that the Bureau of Land Management did not need for the Seeds of Success program would be sent back to the Northwest Oregon Regional Partnership so they could use the seeds in their native nursery. So during my brief time helping with this program, I gathered the seeds of the Nooka Rose, which is on the left, and the Pacific Ninebark, which is on the right. The seeds for the Nooka Rose were collected at Sickasett State Natural Area, while the Pacific Ninebark was collected in Nehalem at the North Nehalem Fish Hatchery. The bulk of my project, so this leads into my, where I did most of my work, was in doing remote work. So this included gathering research and data from multiple sources and compiling it into easy to read maps. These maps were to help create a visual of the locations of area infrastructures and industries, show the habitat types in the county, the movement of fish passage barriers, and where previous projects took place. After the data was compiled, it was saved and shared to TEP via an external hard drive, Google Drive, and ArcGIS. I definitely wanted to make sure none of that data was lost. So on this map, this slide shows one of TEP's programs called the Backyard Planning Program. The Backyard Planning Program is a cost-free voluntary assistance program that private landowners use to remove invasive species from riparian areas to improve habitat for fish and wildlife. This map indicates where the program has been implemented and what years. There is additional data provided that shares the cost of the project, the habitat type, and the agencies involved in the project when you use the map online, and you could click on the stars there. Then another example of the data collection made into map was on the fish habitat distribution and the fish passage barriers within TEP's study area. The map on the left shows an overview of the fish passage barriers in TEP's study area and any known data on those barriers. This included bridges, culverts, dams, tide gates, and more. Then the one on the right shows what else I did with that data. So I was able to split it into whether the passage barriers were passable, partially blocked, or if the status was currently unknown. And then it was color coded like a traffic light where green is passable, yellow is partially blocked, red is blocked, and then the unknown passage status is gray. So this created kind of a visual of the infrastructures that might need our focus in order to restore the habitat of fish in these areas. Then with that data, I also combined it with the fish habitat distribution data from Oregon Fish and Wildlife. There were several fish species I included in this map layer, such as the fall and spring run of Chinook salmon, chum salmon, steelhead, red band trout, coastal cutthroat trout, and more. For the example on this slide presentation, I went with the coho salmon. The map on the left shows the habitat distribution of coho salmon and all the fish passage barriers they might run into in the TEP study area. And then just like the previous slide, I then split it so you could look at just a single type of passage barrier to see where the coho salmons might be impeded by a blocked barrier. So hopefully these maps and data will give a better overview of the area and will hopefully assist in identifying future restoration and conservation needs within TEP's study area. Then sadly, due to COVID, I did not get many opportunities to work directly with the community as I hoped, other than answering questions of people passing by while collecting water quality data or collecting seeds. However, I was able to help volunteer with the native plant sale that TEP and Northwest Oregon Regional Partnership held in October 2020. Northwest Oregon Regional Partnership and Tillamook Estuaries Partnership found a safe way to have their fall native plant sale by advertising remotely, creating online order forms, and organizing a drive-through for the pickup for the public to pick up their purchases. In the advertisements, people were encouraged to consider buying native plants for their gardening by explaining the importance of native plants to the environment. I assisted by directing people to their orders, answering additional questions, and helping load their orders into their vehicles. Additional projects that were done through my internship with TEP included designing a tutorial course where I taught people the basics of ArcGIS Pro and taught people how to use Esri's story map to share information about their work. During the tutorial, I showed the basics of how to set up a base map, add data, and how to create a feature class. A feature class could be a point, the line, a polygon to create a feature on the map. After showing people in the tutorial the basics, I did send them PDFs I created with step-by-step instructions that they could refer to later, and provided additional resources so that they could learn more in the future. A snapshot of the PDF can be seen on this slide. And for the Esri story map tutorial, I showed examples of how story maps are used to share information with a variety of stakeholders and how to add multiple elements to an Esri story map. Additional work and to learn the agency ecosystem, I was invited to a few meetings. I was lucky enough to participate in meetings with people from TEP and organizations and agencies TEP collaborates with. One of the meetings I particularly enjoyed was the one with Nature Conservancy, Tillamook Estuary Partnership, Lower Columbia Estuary Partnership, and Azteca Waters. And in this meeting, they were working to establish a standard method of monitoring on the Oregon coast. So to wrap up this presentation, I just want to finish by answering these questions on this slide. So my hope is that Tillamook Estuary Partnership benefited from my internship as much as I did. At the very least, I hope I contributed as an extra pair of hands to help with the native plant cell, collecting seeds and entering data. I know I benefited by working with Tillamook Estuary Partnership because I learned so much from them. I was able to get some hands-on experience out in the field and had worked from beginning to end on a monitoring project and learned more about using GIS. This has also been a great experience in working remotely and staying motivated to get tasks done. I also benefited by working with Tillamook Estuary Partnership since it allowed me to expand my network as we collaborated with several agencies and individuals throughout this internship project. Throughout my time in the TCBES program and my work with TEP, I was given many opportunities for field experience to gain new knowledge on estuary systems, data management, and so much more. It has allowed me to challenge my own perspectives and best practices for communicating environmental conservation to a variety of audiences. Overall, the TCBES program and my internship project helped confirm my passion and desire to work in estuarine and marine ecosystems. Now, after this project, the plan is to graduate from the TCBES program and continue working on conservation and restoration projects. This summer, I am volunteering with Tillamook Estuary Partnership to get more experience while I start planning my next steps in life. So now that we have come to the end, there are several people I'd like to thank for this opportunity, such as my family and friends, especially my parents, my brother, and my sister-in-law. They all supported me to follow this path, and I really am forever grateful. I would like to thank my mentor, Tamara Enns, who graciously took me on to help create this great project. She was always willing to provide guidance and gave me the ability to jump into any project I found interesting. I really cannot thank her enough for the knowledge that she shared with me and for providing this opportunity. I would also like to thank York Johnson and Mason Miller for allowing me to work with them on several projects, and for everyone else that I worked with at Tillamook Estuary Partnership. They were each so kind and helpful and just provided a wonderful experience. Next, I would like to thank my advisor, Lisa Canelli. She has continually supported me throughout my time in the TCBES program. She has worked tirelessly to make sure I had every opportunity to obtain more knowledge and skills throughout the program. I also could not thank her enough for everything she's done. Lastly, I would like to thank my cohort. Every single person in my cohort is absolutely amazing for the kindness, assistance, time, and memories that they have given me throughout this program. I know that they will each go on to do amazing things in their life. And so with that, with all that being said, I'd like to give a big mahalo to all of you for listening to my presentation. If you need or want to contact me with any questions or comments, my email is listed on the slide as norbaum@hawaii.edu. Well, mahalo.
Thank you, Sarah. That was a wonderful presentation. We'd like to open up the floor now for any questions. So if you have a question and you're shy, please type it in the chat. And if you're brave, please feel free to unmute yourself and ask the question. All right, Dawn, please take the floor. Oh, I think that was, sorry, I think that was a applause and not a raised hand. So Sarah, I do have a question for you. You did a deep dive into the ecological data that was available for your area of interest. And I know you went to multiple sources for those data sets. And I'm wondering, now that you have a better understanding of estuaries, what sort of data do you think still needs to be collected to be able to do analysis of place?
So definitely, sorry if you hear my nephew, he is not having a good time. I would personally think more stuff away from the estuary, because since it kind of all feeds into the area, whether it's out in the ocean or just inland, since it's feeding into it and affecting the waters in that area, any information or data on that would be helpful just to get a better overview of the whole system. Hopefully that answered your question.
All right, so let's turn to our YouTube correspondent. Tracy, how's it looking over there? Are there any questions in the comments?
No, people aren't writing in there.
Okay.
I have a question.
It's easy to monitor, so.
Okay. I heard somebody in the audience say
Oh,
that.
yeah, I have a question for Sarah. With your temperature data, was that sort of a surprise to you and in what you found?
I don't feel like the temperature data was that much of a surprise. Just I think with some of the placement, it was warmer too a little bit in the fall. So I think the surprise for me at least was more the DO levels.
I have a question for you. Sorry, go ahead.
Oh, so my question was based on your water quality data. I'm guessing you're trying to decide whether it would be useful to maybe remove that dike. What do you think? Would that help improve water quality if that effort was made?
I personally think so. Yeah, they have multiple plans right now of whether, I don't think they'll ever be able to truly remove the whole dike, but they have kind of plans to maybe, because if you look at an overview of the map of the area, just the way it flows into the tide gate isn't where it was naturally supposed to flow. So like discussions have been to move it further down to where it should, to create a bigger tide gate, or one of the ones we were talking about as a possibility was like a floating one so that the trail could still be there. So yeah. I think it would help.
The floating idea was to remove the dike entirely and put in a floating bridge so that the tidal exchange was there, but people could still access it. And right now, that's one of the biggest points of opposition, is that people want to be able to walk across the dike the way they have for the last 70 years, and not necessarily recognizing how much that dike has affected the estuary on the off side, because it's changed the dissolved oxygen and the temperatures, as Sarah showed in the data. And Sarah, I have a question about that also. Just what's your opinion about how best to proceed there in order to provide the appropriate temperature and dissolved oxygen for the salmon, which is the primary focus of most of the estuary restorations in Tillamook County? do you think that it would be good to remove the dike, or that shifting the tide gate to its original to the original location of the flow, because they did move, as Sarah said, when they put the tide gate in, they basically put it in a place that is the opposite side of the bay from where the natural primary channel was in the bay. So do you think, what's your opinion on that, removing it versus just shifting it back, or what do you think would be best, or would you like to see?
I would like to see improvement. It's hard to, I think, say exact opinion. When I had talked to Oregon Parks and Recreation Department, it's kind of a controversial subject just because the community is, as you said, kind of opposed to removing it completely for the trail reasons. And then also there's concern since Terra del Mar is kind of built so close to it that it might cause flooding into their homes. So it might not be possible just to remove it completely if we want community support, but maybe just changing even where it naturally flows might increase better, and fixing the tide gate so it's not deteriorating. So it will just kind of increase the dissolved oxygen levels, and then kind of fix the temperature for it.
Right. Well, thank you, Sarah, for your presentation. We greatly appreciate it. And really thank you very much to our three first half presenters. And one of the things I wanted to share with you all is for this cohort, they had plans, right? They were moving forward in the direction they wanted to go, and then COVID happened. And so they each person had to take a pivot on how they were going to manage their work and where they would do their work. So really want to give a shout out to them for their flexibility and their tenacity to make their professional internship happen. So kudos to all 6 of them. And with that said, we're going to go ahead and go into our break, and we'll return at 11:30 for our second half of our presentations. I am going to leave the Zoom up and the live stream on. So if you could please go ahead and just turn off your camera and your microphone. And I highly recommend going outside and saying hello to the sunshine, hopefully, or maybe the rain. I don't know, because I'm in a box with no windows, but I'm going to go find out. So why don't we all go figure out what the weather's doing, and we'll come back at 11:30. So I'll see you all then.
I was asked to unmute.
Hi, everybody. We are not back. We're just going to do a little testing. So you're welcome to stay for our sneak preview.
Kaveh, I don't hear you talking at all. Are you on mute?
Sorry, we're using my audio and I had it
Okay.
off. That's
Can you hear
why.
me now? Okay. I was like, I see her talking, but I don't hear it. I'm a little worried.
I couldn't hear Lisa coming through either.
Because we don't have the output on there. Because
Okay.
it's on mine. Otherwise, there'll be an echo.
You would think we wouldn't have to face all these technological difficulties, but
Here we are.
okay.
We're figuring it out.
So I just wanted to share my screen just to see if everything works. Sorry. Oh my gosh. Okay. How does that look?
We're not seeing anything.
I'm seeing.
I can
You are
see it.
seeing things? Okay,
Yeah,
sorry.
I can see it.
It was my computer.
Okay. Okay, so I can advance. Okay.
Okay, now me.
So there is a bar at the very top that says something about complete setup.
Okay.
Maybe later. Maybe choose maybe later. Okay, now it's gone.
Okay.
I'm going to jump out and jump back in, Kaveh.
Okay, sounds good. Just don't leave me.
I will not. I will not abandon you.
Okay. Okay, is that good, Lisa? Okay, cool. Ooh.
Okay, is it okay if I share back the big screen, or do you guys want to do more testing?
We are good.
Okay, awesome.
Thank you.
Thank you both. Kavehi, even though we can't hear you through that computer, do you mind having the mute button on it? Right, we can't hear you through that workstation, right? But do you mind having muting
So
it?
I haven't joined the audio, so I don't see that.
Oh, that option?
Yeah, it's not
Yeah,
even
that
an option.
Oh, okay, okay.
Is that okay?
Yeah, no, that's totally okay. I didn't want it to accidentally click over to you, but if there's no option, fantastic.
Should be all
To
good.
mute the
Yeah,
audio.
we're all good. Aloha, everyone. Welcome back to the Hilo TCBES Professional Internship Presentation Day event. We are going to welcome our first speaker of our afternoon session, Kavehi Lopez. She is going to be introduced by her mentor, Franny Brewer. Franny Brewer is the Communications Director at the Big Island Invasive Species Committee, also known as BISC. Franny has led the organization's outreach efforts since 2015. She is known for her extensive experience working in outreach and education and her passion about using social science to enhance community engagement in conservation actions. Please join me in welcoming Franny to the Zoom mic.
Thank you, Lisa. I'm so happy to be here today. I'm so excited because I have the honor of introducing Master's candidate Kavehi Lopez today. And Kavehi is a Hilo girl and a graduate of Kamehameha Schools, but she's also a fellow alumni of Hilo. She graduated a few years after me, just a few, but she has her undergrad from Hilo in environmental science. And I first met Kavehi when she was in the PIPES program. She was interning for Stanford University. We were collaborating with a doctoral candidate there on a research project testing social science strategies.
Enter your meeting ID followed by
To encourage community action on invasive species. And at that time, this was a fairly new approach for Hawaii. So it was very new and kind of cutting edge, so to speak, in outreach here. So it was a really intense project. We were working sometimes 6 days a week, evenings, weekends. We were literally meeting with dozens and eventually added up to hundreds of people. And Kavehi was an undergrad intern, but she was amazing. Absolute flexibility. There was a lot of education for each of us on that project because we were all kind of in a new area. Even our social scientist was learning Hawaii and learning about our invasive species issues here. And so everybody on the team was a little bit in new territory. And when you're an undergraduate intern, that can be really disconcerting. And so just seeing Kavehi's amount of commitment and flexibility and consistency coming in every day, committed, prepared, enthusiastic, and really flexible. Like, okay, we're making an adjustment, we're making another adjustment, we're making another adjustment. And she was right there giving feedback, just making these invaluable contributions. So at the end of that summer, when she wanted to stay on the project, she's like, well, I could go into a kupu internship. We were like, yes, please come work for BISC and be our kupu and work on that. So she stayed with us to finish that research project all the way to the very end, right through into publishing. So when a couple of years ago, she approached me about going for her graduate degree in TCBES, I was, of course, thrilled for her, but when she said she wanted to intern with BISC, I was very interested in the opportunities for us, just knowing what an amazing job Kavehi does. So we put together a great plan to do more original social science research. And we were really excited and ready to move forward. And then small global pandemic hit. That has caused a lot of upset for a lot of folks, really derailed our plans. But once again, Kavehi just showed amazing flexibility. And we sat down and talked about, okay, well, we're not going to be able to do this social science project because it's very hard to talk face-to-face with people that you need to be 6 feet away from at all times. So what else does BISC need? You know, she really wanted to make a contribution, like not just check the boxes, but what can I do for BISC that's going to be valuable? And we examined sort of our outreach program, and she looked at gaps and said, yeah, I'm going to work on this and I'm going to fill this. So I'll let her talk about the specifics, but I just want to say that, you know, that's a really big deal when you're setting out on such a big thing as a graduate degree and you're suddenly all of your plans are just ripped to shreds. To be able to bounce back from that and come with a positive idea and want to continue to make contributions it was really impressive. So it's been such a pleasure over the last couple of years to work with her. And this final contribution that she's going to present to you folks today, this isn't, you know, tinkering around the edges. This is a really valuable, huge chunk of our outreach plans going forward. This is really going to be very foundational to our program in the next couple of years. So I really appreciate the work that Kavehi has done. And I want to Mahalo her for all the contributions to BISC and to our team and to me personally. I couldn't have had a more pleasant experience being a mentor. So I appreciate you, Kavehi, and I'm personally grateful that you're here, and I look forward to hearing your work today.
Mahalo nui, Franny, for that amazing introduction. It's been my pleasure to work with you over the past few years. Just hold on a second while I pull up my presentation. Let's see. How does that look for everyone?
Wonderful. Right. Looks good.
Awesome. So aloha mai kākou. I am extremely excited to talk about my professional internship experience with the Big Island Invasive Species Committee. I've titled my presentation, Shifting into the New Normal, Invasive Species Outreach in the Age of COVID-19. And the reason why I included a computer on this slide is because virtually all of the work that I did for my internship was carried out online. So I snapped this picture back in 2019 at our TCBES orientation. This was a week before our first classes started. This Anuenue was absolutely breathtaking, and once I saw it, I was filled with excitement for the next 2 years ahead of me. At this point in time, I had a pretty good idea of what I wanted to focus on for my internship program. But as it turns out, these 2 years didn't play out as I had expected, and I was forced to shift my project plans due to the pandemic. If this experience has taught me anything, it's that I will never really know what kinds of obstacles I'll be faced with in the future. This olelo no eau, aya no ike ko akeau, serves as a reminder for me to live in the present, learn from the past, and do my best to prepare for the future. Before I dive into my presentation, I would like to go over a brief outline of what I will be discussing today. I will start off first with an introduction of invasive species in Hawaii, the organization that I worked with, and then I will discuss the importance of science communication. Next, I will talk about my internship experience, the projects that I worked on, and the deliverables that I produced. Finally, I will discuss the benefits that my work had on the organization, and also how these experiences have shaped my professional path moving forward. Invasive species are federally defined as organisms that are not native to an area and whose introduction causes harm in at least one of 3 areas: the environment, the economy, and human health. The state of Hawaii added to that definition to also include species that impact our way of life. Some of you may be familiar with these particular invasive species, such as rapid ohia death, little fire ants, and the invasive semi-slug, which serves as a carrier for the parasite that causes rat lungworm disease. Hawaii is home to a diverse array of native flora and fauna found nowhere else in the world. And unfortunately, this place is also considered the endangered species capital of the world. Invasive species pose a major threat to the native biota. According to a report published in 2001 by the International Union for the Conservation of Nature, Hawaii has over 50 of the world's 100 worst invasive species. Whether introduced on purpose or accidentally, invasive species possess a similar set of characteristics. The first being that they prey on or outcompete native species for resources. One of the reasons why feral cats are so invasive is because they prey on native ground nesting birds. The second is that invasive species aggressively take over new areas. Fountain grass is definitely one of those species. If you've ever driven on the west side of Hawaii Island, you've definitely seen this plant along the roadsides. Invasive species also quickly mature and reproduce, and rats are a really great example of this. A mother rat can have as many as 8 to 12 offspring about every 30 days, as long as there is enough food, water, and shelter. And young rats reach sexual maturity at approximately 3 to 4 months of age. The last characteristic of an invasive species is its ability to transform landscapes. Rapid ohia death has decimated thousands upon thousands of acres on Hawaii Island, completely transforming forests on Hawaii Island. Those of you that live on the east side of the island are most likely familiar with this plant. Myconia is listed as a state noxious weed and is one of Hawaii's most invasive horticultural plants. It was intentionally introduced to Hawaii Island in 1961, and by the 80s, it was clear that this plant was invading low elevation forests. One of the major reasons why Myconia is so damaging is because it forms thick monotypic stands, shading out native plants and also reducing the amount of rainwater that goes back into the watershed. Their seeds are spread by birds, and also their shallow root systems promote erosion, which degrades the quality of surface water and increases sedimentation of coral reef ecosystems. Before there was BISC, there was the Big Island Melastome Action Committee. It was formed in 1995 with the purpose of controlling the spread of Myconia. It wasn't long before they realized that getting rid of Myconia was going to require a considerable amount of time and resources. The plant was so widespread at this point, and it would be extremely difficult to get it under control. The reason why the response to Myconia was not effective was because there was too little resources, and frankly, we were just too late. However, the lessons learned from Myconia have informed invasive species management tools moving forward. I like to think about it using this invasion curve. So you have time on the x-axis and cost on the y. They always say that an ounce of prevention is worth a pound of cure, and that is definitely the case when we're talking about invasive species. The earlier that we can detect and respond to an invasive increases our chances of eradication. This is where outreach comes in. Typically, most people become aware of an invasive species when its populations are widespread. It is up to us to use the lessons that we have learned to inform how we move forward. The Big Island Invasive Species Committee was formed in 1999, and their mission is to prevent, detect, and control the establishment and spread of invasive species threats to the Hawaii Island environment, economy, and way of life. This picture was taken a few years ago at a staff retreat. The work done at the Big Island Invasive Species Committee focuses on various aspects in the fight against invasive species. We use early detection to seek out newly naturalized plant species along roadsides, and we also use drones to track and identify ohia trees that are potentially affected with rapid ohia death. One of our most recent projects, called Mamalu Poi Poi, focuses on monitoring for Africanized killer bees and coconut rhinoceros beetles at the Hilo and Kona airport. Our plant crew works hard surveying and controlling incipient plant populations or plants that are in the early stages of invasion. Plants like rubber vine, cotton easter, and Moluccan raspberry are some of the plants that we work on. Hopefully these plant names sound unfamiliar to you, and we really want to keep it that way. BISC also serves as a field action team for a lot of the researchers that are working to address rapid ohia death. We collaborate with the U.S. Forest Service, the Pacific Basin Agricultural Research Center, and other agencies to inform the state's management recommendations for dealing with ROD. One day, our crew could be collecting frass samples for pea bark, and another day might be spent collecting seeds for studies on ohia resistance. While invasive species are a threat to our native forest ecosystems, certain invasives also pose a threat to the economy and public health. And albizia is one of those. BISC focuses on addressing the impacts of albizia and works with stakeholders like the Department of Transportation and the Hawaii Electric Light Company to protect both infrastructure and public safety. And last but not least, we have outreach. We do all the typical things that you would think of when it comes to outreach. We create brochures and rack cards to educate the public. We visit classrooms and talk to students. And we also man booths at community fairs to talk to the public. But our outreach at BISC goes farther than that, as Franny mentioned previously. We collect data and use social science to inform our programs so that we can motivate the public to take action on invasive species. Our Little Fire Ant Community Support Program focuses on empowering residents to work together and treat for little fire ants as a collective. While our teams work to address all of these species that I've mentioned, I also wanted to point out how important it is to recognize the reality that new pests are continuing to arrive on our island. In 2002, the Hawaii Department of Agriculture performed a risk assessment on the movement of alien species from the continental US into Hawaii through the Kahului Airport on Maui. The plant quarantine branch conducted 7 3 to 4 week inspections between September 2000 and July 2001. I was in kindergarten at that time. And they looked at checked and carry-on bags and agricultural products that were shipped through air cargo. During those 130 days of inspection, the team intercepted 279 different species of insects, 125 of which were not known to occur in Hawaii. The fact that this study was done almost 2 decades ago means that this number has likely increased over the years. And while not everything arrives and survives long enough to establish and invade, there are a few that slip through the cracks. Here are some of the new invaders that BISC has recently begun focusing on. I want to note that our outreach approach differs from species to species. We are currently working to eradicate 2 new plants, devilweed and phonax, pictured on the top left and right corner. The outreach team is responsible for collecting plant reports from the public and relaying that information to our crews on the ground. The 2-line spittlebug was first detected in 2016 when a rancher in Upper Kona reported widespread die-off of his pastures. While the University of Hawaii's Cooperative Extension is leading the research on the 2-line spittlebug, our role at BISC is to provide them with guidance on conducting outreach with the public. For the semi-slug and rat lungworm disease, we take research from the Jarvie lab at Hilo and the STEM curriculum that they produced, and we deliver that to teachers through a professional development course. The Rami moth is fairly new to Hawaii Island and can decimate mamaki plants. Our role at BISC is to raise awareness about this pest by posting alerts and urging the public to remain vigilant. The Queensland longhorn beetle is another new pest to Hawaii that attacks a number of different tree species, including kukui, citrus, cacao, and avocado. BISC became involved with QLB outreach after receiving a growing number of reports and concerns raised by farmers and other residents. So when it comes to outreach, many efforts utilize what is known as the deficit model of science communication, which operates by taking the science, disseminating it to the public, and increasing awareness around a topic. We utilize this traditional approach selectively at BISC. This is because with some species, we are looking for more action from the public, which requires a more targeted outreach effort. For example, this is the case with little fire ants. BISC created a community support program in order to empower residents with the knowledge and resources to control the ants in their own properties. Research has proven that information-intensive campaigns are minimally effective when it comes to promoting conservation action. And while these campaigns do serve an important role in outreach, when it comes to motivating long-term changes in behavior, we need something more. And so how can we motivate others to take action against invasive species? This has become an important question that drives the work that we do at BISC. Earlier, I talked about BISC's community program, which provides education and support for residents looking to control little fire ants. The creation and delivery of this program was informed by social science and serves to empower and motivate residents to work together with their neighbors to control LFA. My initial plan for my internship was to carry out a pilot study to better understand public perceptions on little fire ant control. My objective was to figure out how BISC could improve their program to better target the needs of Hilo residents. In order to gain a deeper understanding of those community needs, this requires talking to people. And since this project would involve a lot of face-to-face interaction, carrying out this effort was made impossible due to the pandemic. We are very fortunate at BISC to be recognized as essential workers and have been able to continue our field work throughout the pandemic. BISC has implemented new sanitation and social distancing policies, and our crews continue to work hard out in the field. This is a picture of Nate collecting a tree sample to test for rapid ohia death. Since the outreach team had to transition to remote work, my focus shifted towards strengthening BISC's digital presence. Since a lot of our outreach efforts at BISC consisted of in-person presentations and trainings, we needed to make sure that our resources were more digitally accessible. And to do this, we conducted multiple webinars through Zoom and streamed live on Facebook, which covered topics like managing little fire ants, reducing your risk of exposure to rat lungworm disease. I played an important role in the design of these presentations and also created social media posts to promote the events to the public. This is what our staff meetings look like now. We go over project updates and safety briefs, and although we can't be together in person, it's really nice to see everyone's faces and catch up every once in a while. So for my internship, I focused on 3 primary projects. The first was video production, the second was social media campaigns, and the third was a content creation guide. All of these projects gave me the opportunity to channel my creativity and learn more about conducting outreach in the digital realm. So for my first project, I created an 11-minute video depicting the work that our crew does out in the field. This video was made for the Ohia Love Festival, which usually takes place each year at the Iniloa Astronomy Center in Hilo. And this event is really important for us at BISC. In previous years, this event has seen over 1,000 attendees. Since this was the first year that the event was held in a virtual space, our team wanted to come up with a way to create and showcase the work that we do in the field. My intention for the video wasn't to inform the public on all of the different processes carried out in the field, but it was to serve as a source of inspiration for students and other individuals looking to work in conservation. My first step was to come up with a vision for the video and then create a storyboard. I decided to center the video around a set of interview type questions, splicing different video clips and pictures in between. I relied on the field crew to capture the pictures and videos for this project because I wasn't able to make it out to the field with my school and work schedule. This meant that I needed to meet with the field crew via Zoom to clearly convey my vision and my expectations for the video. Once I got all of the footage, I then worked on the editing. And while I had a little bit of experience with iMovie, I challenged myself to use a new editing software called Final Cut Pro. And this program wasn't as intuitive as I would have liked, so I spent a lot of time watching tutorials on YouTube. And after many hours of tutorials and editing, I finally finished the video. I created a promotional graphic to share on social media, as well as a 1-minute trailer. So this video made its premiere at the Ohia Love Festival week-long event. We hosted a live webinar on Zoom and Facebook. We showed the video and then we hosted a live question and answer panel with our ROD crew. This gave the team a chance to talk with the public and also give them a better idea of who they are and the work that they do out in the field. The event was a success and we had about 77 people show up for the webinar. After showing the video, we did a question and answer session, which lasted around 40 minutes. If anyone is interested in viewing the webinar presentation, it was recorded and can be viewed on BISC's YouTube channel. And also, if you're interested in watching that 11-minute video, please check out BISC's YouTube channel. The next project that I focused on was social media. I worked on 2 separate campaigns. The first was Stop the Ant Month, which happens every October. And the second was Hawaii Invasive Species Awareness Month, which takes place every February. This was the first time that these events were held in a virtual environment. And for those of you who aren't following us yet, I included our social media handles down below. You can find us on YouTube by searching BISC, B-I-I-S-C, and we're on Facebook at UH-BISC and Instagram at Big Island Invasive Species. Traditionally, Stop the Ant Month would involve a bunch of community presentations and hands-on trainings. It also involved school visits and activities with students. And all of this was sort of off the table once the pandemic hit. So in order to keep these events going, we had to figure out what Stop the Ant Month was going to look like moving forward. I was designated as the organization's point person for Stop the Ant Month, which meant that I had to attend planning meetings and collaborate with people from multiple agencies and organizations across the state. Our main objective was to figure out how we can effectively deliver our messages in an online environment. From creating social media posts to posting blogs to doing virtual class presentations, we really had to figure out what this event was going to look like. I designed 23 graphics for social media and also managed the BISC Facebook and Instagram. I also worked with Jamie of the Oahu Invasive Species Committee to manage the Stop the Ant social media pages as well. Here are some of the posts that I created for Stop the Ant Month. I found it very helpful to look at our social media analytics afterward to see how our posts were performing. I noticed that certain posts would do better than others, and that differed between platforms. For example, I experimented with animated posts like the one on the top right. After looking at the analytics, I realized that while it performed well on Instagram, it didn't receive as much engagement on Facebook. Another thing that I noticed was that our followers on Instagram were of a younger demographic than those on Facebook. This ultimately meant that in order to drive up our engagement rate, we really needed to come up with targeted strategies for each platform. I took what I learned from Stop the Ant Month and applied that to my work for Hawaii Invasive Species Awareness Month, or HISAM. One of the main objectives of this outreach campaign is to raise awareness of invasive species issues across the state. This event happens during the legislative session, which makes it a great opportunity to raise awareness and garner support for biosecurity and other invasive species legislation. Our outreach team decided to launch an interactive campaign called Which is the Worst? And we encouraged our followers to cast their vote for the worst invasive species on Hawaii Island. The idea for the game had been suggested at one of the HISAM planning meetings, but no one really knew how to implement it. I decided to take on the challenge and thought it was a good way to engage people in conversations about invasive species. It would also create space for people to share their experiences and to make people feel like their opinions matter and that we're listening. Outreach is not a one-way street, and it's essential for us to be engaged in these conversations with the public. And in this day and age, people are having more and more conversations online. And as an organization, we have to figure out how we can cater our outreach to reflect that. So every other morning, 2 invaders went head to head, and our followers got the chance to vote for the one that they thought was worse. I used the polling feature on Instagram stories to make it easier for people to participate in the voting process. It also made it easier for me to track the results. On Facebook, I noticed that most of the voting took place within a photo album named Hi Sam. Individuals submitted their vote in the comments section. Other members of the planning committee were considering adopting this approach for future events, and so this campaign was closely followed by other conservation organizations. And while I was unsure of how everything was going to play out, this game turned out to be a hit. Throughout the month of February, BIS gained 137 new followers on Instagram and 61 new followers on Facebook. Here are some of the graphics that I created for Hawaii Invasive Species Awareness Month. All of these posts performed really well on Facebook and Instagram. Rapid Ohia death was chosen as the worst invader on Hawaii Island. And although putting this game on was hard work, I really thought that it was an effective way to connect with our audience. In addition to the game, BISC hosted 3 webinars throughout the month of February. I chose to create individual graphics to announce each event and made sure to include the Zoom links directly in the social media posts. We found this strategy to be very effective, and our webinars at BISC had the highest number of attendees out of all of the other webinars during the month. Other posts that performed well were memes and pest alerts. Social media analytics are really important in this regard because they provide data on what kinds of posts are resonating with our audience. And this really helps to inform our efforts moving forward. The last project that I focused on was creating a digital communication plan for BISC. I really wanted to take what I learned from the previous social media campaigns to create something that was going to help the outreach team moving forward. Before this, BISC had no formal social media plan, and so it was important for me to provide that structure. I figured I'd share just a few pages with you all to give you a better sense of what the document looks like. The first half of the plan includes an overview of basic principles of graphic design, as well as tips and tricks for working with Canva, an online graphic design program. One of the most important things that I learned from working on Stop the Ant Month and Hi Sam was that posts that do well on Instagram don't always perform well on Facebook. And so it's essential to understand how we can create targeted content that performs well on both platforms. I did a bunch of research on the Instagram and Facebook algorithms for 2021 and provided the team with tips on how to increase their engagement and reach. The last half of the plan consists of a content calendar for the rest of the year. I outlined the focus for each month, main outreach messages, and provided ideas for graphics and other forms of content. And the following page is a calendar to plan a monthly posting schedule. This living document provides a plan for creating and scheduling content throughout the rest of 2021. I wanted to touch on some other experiences that have contributed to my growth as a science communicator and outreach professional. Anytime that I was assigned a project for any of my classes, I focused on creating products that would be useful to the Big Island Invasive Species Committee. For my qualitative research methods course, I carried out a research project to understand how little fire ants were impacting surfers. I got to practice 3 different research methods, including interviewing, surveying, and participant observation. During my GIS course this semester, I've been working on creating maps that can be used to communicate with legislators. Our internship course with Lisa Connelly last semester was geared towards learning about new science communication tools. We got to create an ArcGIS story map, which allows you to build a web-based story with text, maps, and other multimedia content. We also got to learn about podcasting. I got to interview Carolyn Wong from the Natural Resources Conservation Service in Kamuela about her work on the 2-line spittlebug. I had a lot of fun working on this project and could definitely see myself making more podcasts in the future. I was also introduced to the world of TikTok during Lisa's class. And while I don't see myself creating a BISC TikTok account anytime soon, I do understand the importance of integrating short form videos into our outreach efforts. One of the coolest things about the RAD video is that it continues to be used by BISC for our school presentations and career days. This project also taught me how videos can serve as an important outreach tool. I was introduced to Canva at the beginning of 2020 and really used my internship to learn more about graphic design. Previously, the BISC team was using Adobe InDesign, but that program is expensive and didn't really allow for collaboration. And so I proposed that BISC discontinue using InDesign and switch to using Canva. Now it takes less time to create graphics and we can collaborate on projects more effectively. The digital communication plan that I created serves as a living guide for the outreach team and will inform their campaigns moving forward. And although my initial internship project plans changed due to the pandemic, I got the opportunity to work on projects that ultimately benefited the organization. And as my internship was coming to an end, I asked my mentor, Franny, if they had the funding to hire me on after graduation. Thankfully, and due to the hard work of our program manager, Springer K, they were able to secure funding, and I'm slated to start working full-time with BISC in July. They are currently in the process of developing a brand new position that will reflect a lot of the work that I completed during my internship. My experiences have taught me about the power of narrative, and moving forward, I'm really excited to hone my skills as a storyteller. Building off of that, my internship has allowed me to gain a deeper understanding of the value of photography and videography for conservation. And moving forward, I plan to learn more about digital media production. The last thing I have on this list is grant writing. And so this is something that I've been wanting to learn for the past few years, but unfortunately didn't get a chance to do that during my internship. Moving forward, I really look forward to gaining experience with grant writing. And with that, I'd like to give a huge shout out and mahalo to Springer, Franny, Molly, Jade, and the rest of the Biscuits for their support throughout my graduate school journey. I would also like to thank the Hau'oli Maoloa Foundation, the Office of Hawaiian Affairs, Kamehameha Schools, the Big Island Association of Nurserymen, as well as the Richard Smart Ohana for making it possible for me to obtain my graduate degree. Lastly, I would like to thank Lisa Canali for her guidance and support, and the rest of the TCBES cohort for their friendship over the past 2 years. Mahalo nui loa, and I would take any questions that you guys have.
Thank you, Kafehi. That was fantastic. So if you're brave, please feel free to unmute yourself. We have time for like one question before we're going to take a short break and bring on our next presenter. So, or if you're shy, you can type it into the chat. All right. So Kavehi, I do have a question for you, and it's related to the video production work. And so I'm wondering, in terms of that deep dive that you want to do with video production, like where do you see yourself going? Are you thinking about maybe making a short documentary, or like what's your vision for your video venture?
That's a great question. Thanks, Lisa. In the next few months, we'll be working on, at BISC, a new outreach campaign focused on providing the community with guidance on controlling common weed species. And so we're going to take a lot of the research that we've done internally and create content that's easily digestible by the public and really easy to follow. And so in the next few months, that's what I'll be working on.
So your mentor would like to know what is, in terms of the next big thing for communications and graphics at BISC, is there going to be, do you think there's an area that they aren't exploring right now that you would like to bring them into?
Thanks, Franny, for the question. I think that videos are really powerful in this day and age, and it's something that we could be utilizing more at BISC. And so when it comes to creating content, I think that we need to integrate more of that into what we do. It's a way to create more of a personal connection with our audience, and I think it's going to be really useful moving forward.
All right, thank you. So Becky, she's our YouTube correspondent for the afternoon. Do we have any questions on the live stream? Nope. Okay. All right. So I think we'll just do one more quick question. And this is a question that says in the chat that says, are there other elements of an online presence plan that you're hoping to incorporate?
Thank you, Kylie, for that question. One thing that we've been talking about, especially at our last meeting, was incorporating live videos into our outreach. So one thing that I think we could be doing better is going live more and just creating more opportunities for people to hear our voices and to create that connection.
All right, well, thank you once again for your wonderful presentation. It was beautiful. Which we would expect nothing less, right? Because you're our graphics expert. With that said, we're going to go ahead and take a 4-minute break. We will come back at 12:15 for our next speaker of the afternoon, Joffrey Grimmett. So we'll see you all in a few minutes. All right, thank you all for joining us again for our 5th presentation of the day. Our presenter will be Joffrey Grimmett. He'll be introduced by one of his project mentors, Dr. Diana Felton. Dr. Felton received her MD at the University of California at Davis School of Medicine in 2006. She is currently the state toxicologist at the Hawaii State Department of Health's Hazard Evaluation and Emergency Response Office. At the HERE office, she works with many state and community partners on issues such as childhood lead poisoning prevention, safe fish consumption, air pollution risks, pesticides, and other environmental health hazards. Fantastic work, we need it. And so without further ado, I'd like to welcome Diana Felton, Dr. Felton, to introduce Joffrey.
Hi, everybody. Thanks so much for having me today. It is such a pleasure to be able to introduce Joffrey today. He spent his internship with us last summer as part of the Hawaii Childhood Lead Poisoning Prevention Program, which we affectionately refer to as HICLIP. And HICLIP is a CDC-funded program that is run through the Department of Health through both the Children with Special Health Needs branch and my office, the Hazard Evaluation and Emergency Response Office. So the combination of groups really allows for a really interesting approach to environmental health and prevention of a major environmental health program. So Joffrey just did an excellent job, which he'll tell you all about, but we were just so pleased to have him. Unfortunately, with COVID restrictions, we didn't get as much one-on-one in-person time as we would have liked, but he just did an amazing job taking a really complex problem and producing incredible evaluation and very informative work that's going to help inform the program for going forward. So without further details, I'll let him explain his wonderful project, but we're just so grateful to both Joffrey and the program. And I also wanted to take a moment to recognize 2 other HERE staff members, Casey Ingalls and Kelly Hoffman, who were instrumental in assisting Joffrey with the project as well. So thank you so much to Lisa and the group, and especially to Joffrey. Go ahead, Joffrey.
We can see it, but I can't hear you.
Can you hear me now?
We can hear you now, but we can't see your slides.
Oh yeah, because I stopped sharing, sorry.
Oh, okay.
Yeah. Okay. So again, thank you, Diana, for that great introduction. And aloha, everyone. Thank you for attending my TCBES professional internship presentation, where I'll be discussing the internship that I performed at the Hawaii State Department of Health's Hazard Evaluation and Emergency Response Office. And the title of my talk today is The Contribution of Lead Contamination Sites to Childhood Lead Poisoning in the Hawaiian Islands. However, before I get into the details of my project, I want to just give you a brief outline of what my talk will cover today. First, I'm going to go over the background and purpose of my internship project and my internship agency. Then I'll cover the strategies and methods that I used throughout my internship. Next, I'll present the outcomes of my project, specifically the deliverables that I was able to produce. And finally, I'll discuss how this internship has helped inform my professional path and future. So as you all know, lead can be very detrimental to a child's health, where exposure can lead to a wide array of cognitive and behavioral deficits, such as difficulty paying attention, decreased IQ, and poor academic performance, along with the other effects that are listed in the purple circle shown in this diagram, which, as you can see, also causes a ripple effect that follows a child into their future by affecting their employment prospects, their ability to socialize, and by increasing the likelihood that a child will experience lifelong health problems in general. Which all of this then culminates with costs at the societal level, where unemployment, juvenile justice, and healthcare are all negatively affected. So now comes the question of how does a child get lead poisoning? And the answer to that question is that a child usually comes into contact with a lead exposure source in their immediate environment, where the majority of exposure in the United States is due to deteriorating lead-based paint on the interior and exterior buildings. And the reason why children are at particular risk of exposure when it comes to lead-based paint is due to them participating in exploratory hand-to-mouth behaviors, which can result in them ingesting lead-contaminated dust and paint chips. Soil may also be a source of exposure due to becoming contaminated by exterior lead-based paint, or it can also become contaminated through the historic use of leaded gasoline, welding, and auto repair. And finally, some other risk factors that are specific to or more common in Hawaii are the smelting of lead fishing weights, the use of certain religious and cultural products, and volcanic emissions creating acidic conditions that cause home water catchment systems to leach lead from roofing and plumbing materials. Okay, so now if a child has come into contact with an exposure source, the main indicator of whether a child has lead poisoning is a child's blood lead level, where a blood lead level that is greater than or equal to 5 micrograms per deciliter means that a child has lead poisoning. Though luckily, as time has gone on, we've seen blood lead levels continuously decrease with the introduction of policy aimed at mitigating and preventing lead poisoning, which, as you can see in this figure, starts all the way back with the Lead-Based Paint Poisoning Prevention Act of 1971 and goes all the way up to the lead, dust, and soil hazard standards that were established in 2001. So overall, this observed decrease has been a huge public and environmental health success. However, in recent years, there's been a slight rise in the 95th percentile of blood lead levels for children aged 1 to 5. This rise in blood lead levels has also been seen in Hawaii, where elevated blood lead levels have ranged from 0.8 to 1.4% of the total number of children tested for the years of 2013 to 2019, which you can see in the bottom row of this table, which also presents another problem. Testing rates in Hawaii are very low, where less than 16% of children were tested in the years of 2017 and 2018. So there's a decent possibility that some cases may have gone undocumented. All of this then leads us to the purpose of my internship, which was to aid in the monitoring and the prevention of childhood lead poisoning by assessing how environmental factors, specifically lead-contaminated sites, may be contributing to childhood lead exposure in Hawaii. And I performed this internship at the Hawaii State Department of Health's Hazard Evaluation and Emergency Response Office, whose mission statement is to protect health and the environment by providing leadership, support, and partnership in preventing, planning for, responding to, and enforcing laws relating to releases or threats of releases of hazardous substances to the environment. The HERE office fulfills this mission statement by documenting blood lead test results, following up with a child's physician in order to coordinate treatment, providing lead site investigations at a child's residence to identify the source of exposure, and by providing informational guides to concerned parents. Finally, this internship also allowed me to collaborate with the Hawaii Childhood Lead Poisoning Prevention Program, which is a state-based program whose goals are to increase identification of children with lead poisoning and to coordinate surveillance, clinical treatment, and environmental services. Okay, so now let's get into how I tackled this issue with my project. So my approach basically consisted of 5 overall steps, where the first step was to obtain the residential addresses of children who received a blood lead test between the years of 2015 and 2019 from the Hawaii Electronic Disease Surveillance System. Next, I used GIS to map residential addresses with their corresponding blood lead level. Then I pulled environmental pollution data from the state's IHEAR and Access databases. I followed this up by then creating a layer of IHEAR and Access environmental exposure sites in GIS. And finally, I assessed how these environmental factors may be contributing to childhood lead exposure in Hawaii. Okay, so now we're going to get into more of the specifics of each of those steps. So as I mentioned in the previous slide, blood lead test results that occurred between the years of 2015 and 2019 were chosen for mapping and analysis. This initially consisted of about 81,204 cases. Test results that were not from Hawaii, did not have a residential address recorded, possessed an incomplete address, or only had a medical provider's address were then removed, which resulted in 41,436 cases being left for mapping and analysis. Test results were pared down again after the removal of cases that only contained a post office box as an address. And the reason why post office boxes were removed was due to prior studies which concerned environmental effects on blood lead levels, stating that exact locations were better for statistical analysis rather than PO boxes. And this was due to exact locations being more representative of a child's immediate environment. So after this, test results were then filtered to only include children less than 6 years of age, which left 28,372 cases for mapping and analysis. These remaining test results were then geocoded to latitude and longitude coordinates using the United States Census Bureau's geocoding tool. So when it came to the environmental exposure sites that I mentioned before, these sites were divided into 3 categories: confirmed sites, potential sites, and well sites. And 2 important tools that I used to identify those sites are the state's IHEAR and Access databases, where IHEAR is a map-based database that catalogs known sites of environmental contamination across Hawaii, which would include public, commercial, and private locations. It's managed by the HEAR office and contains geographic data, history of remediation work, and other pertinent documents. Similarly, the Access database also catalogs similar information. However, it's an older database that's mainly for internal use. And the main reason why I use it was that it contains some sites that have yet to be transferred over to the IHEAR system. So going back to the sites, the first category of sites, the confirmed sites, were identified using the IHEAR and Access databases. And the criteria that I used to label a site as a confirmed site as opposed to a potential site included whether the site was suspected to have some form of lead contamination based on historical use, and the site having analytical results that confirmed lead was present at the site with concentration values greater than or equal to the Department of Health's environmental action level of 200 parts per million for lead in soil. Also, you can see an example of a confirmed site on this slide. This is Factory Street, which is located in Kalihi over here on Oahu. And Factory Street is probably considered the most famous lead contaminated site in Hawaii. And this is just due to the very high lead concentration values that were present at the site. They go up to about 121,000 ppm. And then also just the long journey that it took to get this site cleaned up. It took about 27 years for abatement to have occurred and to just be finished, which that took so long mainly due to just issues over jurisdiction over the site. Potential sites were also identified using the Hawaii State Department of Health's IHEAR and Access databases. However, some bridge and airport sites were identified using the Historic Hawaii Foundation's 2013 bridge inventory and the Hawaii Department of Transportation's list of abandoned and forgotten airports located on the HDOT website. And the criteria that I used to label a site as a potential site as opposed to a confirmed site was whether the site in question was suspected to have some form of lead contamination based on historical use, but did not have confirmatory lab results that showed lead contamination at or above the Department of Health's environmental action level of 200 parts per million for lead in soil. So onto the final category site, which were the well sites. So initially, we were just going to compare elevated blood lead levels with confirmed and potential lead sites. However, Diana came across an article in PNAS that found that children in North Carolina that relied on private wells for drinking water had a 25% chance increase in odds of having an elevated blood lead level, as opposed to those who got their drinking water from sources that are regulated under the Safe Drinking Water Act. And the suspected cause of this increase was likely due to household plumbing and well components corroding. So given that there are a multitude of homes across the islands that rely on wells as a source of drinking water, we decided to add these sites to my map into my analysis. These well sites were then identified using a GIS layer provided by the Department of Land and Natural Resources Commission on Water Resource Management, which initially included 5,244 wells. But since the study was only concerned with wells used for domestic drinking purposes, wells that did not fit this criterion were not included in mapping or analysis, which resulted in 899 wells being eligible. And now for the final step, we used the compiled data to make two maps, one that contained elevated blood lead levels in site locations, and one that contained elevated blood lead level rates for census tracts and the location of sites within those tracts. Both of these data sets were then analyzed using a multiple linear regression model, which, if you're unfamiliar with, is a statistical model that is used to estimate the relationship between two or more explanatory variables and one response variable. So for the first map, we use this type of model to determine whether there is a significant relationship at the state and county level between blood lead levels and distance from nearest site and lead concentration value at the nearest site. And for the second map, we determine whether there is a significant relationship between elevated blood lead level rates for census tracts and their number of sites within those tracts at both the state and county level. Okay, so moving on to the outcomes of that mapping and analysis. So here's a statewide view of the first map that contains elevated blood lead levels in site locations, where you can see elevated blood lead levels are represented with an orange dot on the map, where increasing size of the orange dot corresponds to increasing elevated blood lead level values. Confirmed sites are then represented with a black dot on the map, where increasing size of the black dot corresponds to increasing lead concentration values at the confirmed sites. Then potential sites are represented with pink dots, and finally well sites are represented with blue dots. Also, before I move on, I should also mention that the orange dots that you see on the map, or the elevated blood lead levels, have undergone a process called jittering, which basically means that I added some random noise to those locations. And the reason why I did this for this presentation was to keep the protected health information and other private information of those patients confidential. So the multiple linear regression model at the state level showed a statistically significant relationship between distance from nearest potential site, where the median blood lead level increased as distance between nearest potential site doubled, which, as you can guess, was odd to us since we expected the opposite to happen. So one possible reason that I thought of to explain this occurrence was just the possibility of lead being absent altogether when it came to some of these potential sites. And again, this was just due to a lack of analytical data on those sites. Finally, the multiple linear regression model at the state level also found another statistically significant relationship between blood lead levels and lead concentration value, where median blood lead levels increased by 0.27% as lead concentration value doubled. Moving on to the county level, the multiple linear regression model for Honolulu County yielded a statistically significant relationship for distance from nearest confirmed site, where median blood lead levels decreased by 1.32% as distance from confirmed site doubled. There is also a significant relationship between blood lead levels and distance from nearest potential site, where median blood lead levels increased by 0.88% as distance from potential site doubled. And finally, there is a statistically significant relationship between blood lead levels and lead concentration value, where median blood lead levels increased by 0.38% as lead concentration value doubled. For Hawaii County, the multiple linear regression model yielded a significant relationship between blood lead levels and lead concentration value, where median blood lead levels increased by 2.2% as lead concentration value doubled. Next, the model for Maui County showed no statistically significant relationships between any of the variables. And Kauai County would show similar results with no significant relationships found between any of the variables. Now, moving on to the second map, which shows elevated blood lead level rates per census tract along with site locations. So as you can see, confirmed sites, potential sites, and well sites are represented in the same way as they were with the first map. However, increasing elevated blood level rates are represented with increasingly darker shades of color. So as you can see, it goes from beige to dark red. So the statewide results show a statistically significant relationship between elevated blood level rates and number of well sites within a census tract. For median elevated blood lead level rates increased by 27.59% as the number of well sites doubled. Moving on to the county level, I saw no significant relationships between any of the variables. Oh, sorry, that was for Honolulu County. Next, for Hawaii County, the multiple linear regression model did not yield any significant relationships between any of the variables. Maui County would continue this trend with no significant relationships being found between any of the variables. And finally, Kauai County also did not yield any significant relationships between any of the variables. So to summarize, overall, our results did not demonstrate a relationship of elevated blood lead levels with residing closer to the confirmed or potential lead contaminated sites that were identified using the IHEAR and ACCESS databases. This supports the idea that most childhood exposures to lead in Hawaii are occurring from lead contamination in and around the home. Also, when it came to elevated blood lead level rates within census tracts, we found that there was a relationship between elevated blood lead level rates and the number of well sites within a tract. This finding is of interest as drinking water is rarely considered a likely source of lead exposure in Hawaii and will require further investigation just due to possible confounding variables, especially since this relationship was not found at the county level. Okay, so now I'm just going to go over some limitations that I experienced during this project. So one limitation that I experienced during the study was the limited quantity of test results due to Hawaii not currently performing universal testing when it comes to blood lead tests. Also, since children are tested for lead only if they screen in for being at risk, the children getting blood tests for lead have a higher pretest probability of having an elevated blood lead level. Therefore, the dataset does not represent all children in Hawaii. Additional limitations of the available data include test results with incomplete address information, results only having PO boxes or healthcare provider addresses, and the fact that not all eligible test results were available to be geocoded. These limitations resulted in there only being 28,372 test results that were available for mapping and analysis. Another possible limitation has to do with our choice of using the largest test result available for cases that had multiple test results. And this was regardless if the test result was from a venous or a capillary blood lead test. And the potential issue with this decision is that there's a significant rate of false positives there's a significant rate of false positive results from capillary lead tests, creating a possibility that we included false positives in my analysis. That I included false positives in my analysis. And one final limitation was the search limitations for both the IHEAR and ACCESS databases, where it's possible that some sites may have been excluded from the analysis due to neither system possessing the functionality to search by contaminant or by substance. So some ideas that I thought of that could be used to expand upon this project in the future were to include the lead concentration values for private domestic wells in future analyses, and to include housing information such as age of housing in order to assess its effect on both blood lead level values and elevated blood lead level rates per census tract. Okay, so now moving on to more general outcomes. So one of the goals of TCBES professional internship program is for the intern to participate in meaningful and challenging work. And I feel this is especially true for my internship, since having the opportunity to accomplish work that will help aid in childhood lead poisoning prevention in Hawaii has made this internship just both meaningful to myself and to the community as a whole. And also, since lead has been known to negatively affect both animals and vegetation by inhibiting an animal's ability to synthesize red blood cells and by stunning plant growth, this work will also be incredibly significant to the environment, since the compiling of this data could lead to future abatement efforts that could help mitigate lead poisoning incidents for those organisms. The internship also constantly provided new and exciting challenges, such as learning GIS, which I had never even used, let alone seen before. Also, putting the skills that I just learned, putting the RStudio skills that I just learned in my quantitative ecology class to the test, and the process of compiling all of my work in order to present it at the end of my internship to both the HERE office and to the Hawaii Clip team was just a satisfying and positive experience. During my internship, I was also given some auxiliary tasks, which will have a huge benefit to the HERE office and to Hawaii Clip. One of these tasks was the creation of a high-risk map based off of census data, such as sociodemographic and housing risk factors, which allowed us to identify areas where children may be at highest where children may be at highest risk for lead exposure. Another benefit provided by this map was the ability for the Hawaii Clip team to compare it to a previous high-risk map that they had made, which was more so based off of zip codes, where this comparison will potentially allow the HERE office and the Hawaii Clip team to construct a more comprehensive high-risk system. One other event that I was also given a chance to participate in was a lead site investigation, where we took soil, paint, and dust samples at a residence in order to identify possible sources of lead exposure. And this was for a child who had tested for an elevated blood lead level. And this experience was just really cool because it allowed me to see more so the direct effects of both my work and the HERE office's work and trying to prevent like this child from, being exposed to this lead source. Additionally, another goal of the TCBES professional internship program is for the intern to become fully integrated into their agency, where they're comfortable with the culture of the agency and their policies. And due to the welcoming demeanor and tutelage of my mentors, I was able to seamlessly be integrated into my agency, where I feel the results of this easy transition can be seen in my work, where I was able to collaborate with a multitude of my colleagues, which undoubtedly benefited the project as a whole. And this was just due to having like a diverse set of voices that provided me with needed advice, critique, and ideas that helped me make this project as comprehensive as possible. So to conclude, my TCBS journey has been incredibly invaluable to me in my future endeavors. The program has been very accommodating and supportive of me when it's come to my interest in environmental health and epidemiology. My professors and colleagues have also taught me so much about the conservation and environmental science fields in general, which has just expanded my knowledge and awareness of fields that I had no prior experience in. My colleagues have also been really important to me since they're my biggest source of inspiration and have been a great support system throughout my entire time in this program. And of course, my internship was also an incredibly fulfilling experience that allowed me to gain a deeper insight into the environmental health field, while also being able to participate in work that will help make a difference in Hawaii. And I'm particularly grateful to my mentors who were extremely kind, informative, and supportive of me and my goals. they made a constant effort to make sure that I had an opportunity to be involved with as many different facets of their work as possible. And I'm really hopeful that we'll be able to work again with each other in the future and possibly expand upon this project. So overall, my time in this program has been really special and will continue to be influential in my professional path and future development. And finally, I'd like to again thank my mentors, Dr. Diana Felton, Casey Ingalls, Kelly Hoffman, and the entire Hawaii State Department of Health's Hazard Evaluation and Emergency Response Office, along with the Hawaii Childhood Lead Poisoning Prevention Program, for providing this internship opportunity to me and for their guidance and support throughout this entire process. I'd also like to thank my advisor, Lisa Canali, for her assistance in obtaining this internship and for her constant support over the course of these past 2 years in the TCBES program. And finally, I'd like to thank the University of Hawaii at Hilo, the TCBES graduate program, and my cohort for making these past 2 years of graduate school a very special and fulfilling experience. And that was my presentation. Mahalo to you all. Thank you. If you have any questions that I'm not able to get to, my contact information is at the top right of the slide.
All right. Thank you so much, Joffrey. That was very informative. Greatly appreciate your work on this and your willingness just to jump in and do a bunch of self-teaching. Your maps looked great. So I'd like to open up the floor for questions. So if you are brave, please just go ahead and unmute yourself, or you can place it into the chat. And while we're waiting for that typing to happen, I have a question for you, and that's related to that deep data dive that you did to into those two databases. And you know, I'm wondering, now that you have seen like how much data needs to be cleaned up, and you've recognized what fields maybe were missing, like what suggestions would you have to the HERA office in moving forward in terms of their data collections?
Well, I know that currently, or they're in the HERA office was in the process of updating those databases to where they would be more, they would have the ability to search by contaminant or by substance. So I think that's a huge step, because that would really cut down the time to, search through all those documents. But also, I think for the most part, all of that data was pretty well organized and kept together. The only thing that how should I say, that was a little bit hard was sometimes it wouldn't have like coordinates or anything, or like an actual location. So I would have to, do some digging, but that was never really a huge issue. It was all good.
I have a question.
Yes.
Since you didn't find that the distance was really important, but and you think most lead exposure is coming from the home, what would be like the next steps to try to like pinpoint what are some of the factors? Would it be like looking at household income, or some kind of neighborhood statistics, or what kind of things would you want to know to kind of pinpoint what are the reasons, age of the houses in the area? What are the things you want to kind of learn next? Would you suggest?
So I think age of housing is really important, because again, like the older the house, the more likely it is to have lead-based paint. I also think sociodemographic like information, like you said, as like income, whether that person is making, below the poverty line or stuff like that. Also, I guess just age of the tenants too, like could also help out, just because like someone who's younger is probably going to, I guess, more so have the funds to, only have the funds to maybe rent or probably just rent like a place that may be contaminated with lead.
Right, thank you. So Becky, is there anything happening on the YouTube channel? Nope, no questions through YouTube. All right, so I'm going to give you all a couple more minutes to think of a question, and feel free just to go ahead and unmute yourself.
Hi, Jeffrey. My name's Lenore. I have a quick question for you regarding the last map that you created about, maybe you could actually flip black in the slides. So it was that one there, the high risk map.
Yes.
Can you talk about how you created that and what social vulnerability index means?
So the social vulnerability index is like a metric used by the CDC that takes a bunch of risk factors such as, again, it's like sociodemographic information. So it's income, it has housing risk factors like age of housing, it has minority status, it has level of education, it has all those things. Um, and what what one of my mentors did, Casey, she was the one who calculated the weight one value that you see here, which is what we use to basically symbolize risk or represent risk or increasing risk. So I basically just took that data and then made this map. And then the main thing that the main things that I did was just noting where differences were in like this map and then the previous map that Hawaii Clip already had. So like some of the differences were like with this map, it had North Kona and the Hamakua area as high risk, whereas the previous one did not. I believe it was not considered high risk. So it was just pointing out those types of differences, and then I presented that to the Hawaii Clip team during their June coalition meeting.
Awesome. So just a follow-up question then from that. Did you plot the actual cases over the top of this map?
No, I did not. This was just seeing like where new high risk areas were and where they had previously been before, which is what the black dots symbolize on here, which were the previous high risk zip codes from the other map.
Okay. And then just I'm sorry to keep asking so many questions, but
No.
the weight numbers there, can you help me understand what they mean, what the differences are? I mean, numerically, I can understand the difference, but if you could just talk a little bit about, you know, these decimal points don't really mean anything to me. So if you could help explain that, that would be helpful.
Okay, so the reason why it's spaced out so much was just to, I guess, be as granular as possible, just so that we can just have a, let's see, sorry, I'm trying to think about the word is, just to break it down as much as possible to see like where, just to highlight what the highest risk areas are. So the the divisions that you see here are, I don't want to say arbitrary, but it was more so spaced out like that, again, just to have that like stepping scale, so we could have some differentiation and to see like what areas should be, you know, narrowed down so that they can be prioritized, you know, for possible, you know, lead poisoning prevention efforts like abatements, or, you know, like giving out the questionnaire to children in that area, which the questionnaire is used to target lead tests in Hawaii. That's how we do that.
I see. So still a follow-up question. So let's just use Big Island for an example. Is it at a
Mhm.
higher risk because of older housing stock, or is it a combination of things like socioeconomic status? And I mean, can you pull that out from this map, or are we just looking, when we're looking at it all together like that, it's difficult to know why there are these differences? Do you have any insight into that?
Um, I'm I'm not. Mm, I would agree that it's probably going to be a bit harder with this current map as to pull that specific information out. But um, if I had to make just a guess or to give like my opinion, I would imagine it has to do more of just older housing. But again,
Okay.
like I can't be 1,000% sure about that.
Right. Sorry to keep putting you on the spot. I just have lots of questions, but
Oh
I
no,
saw
it's
Diana just posted the vulnerability index from the CDC, which is really helpful. So I'll take a look at that. Thank you so much, Jeffrey.
thank you for your questions.
And I can just chime in a little bit about the SVI. So the CDC's SVI, which is what Jeffrey used for this, and Casey, is sort of designed to try to get a picture of communities that are at higher risk from external stressors from a lot of different things. So in the past, a lot of look at environmental disparities or health disparities has all been based on income, and the SVI is designed to look at more than just income. And so there's a whole list of criteria. And so what was so useful about doing this project was in the past, our high risk zip codes, which are represented by the black dots that High Clip has used in the past, are really based mostly on income and age of housing and sort of lower economic neighborhoods. And we wanted to get a broader look at other things that may be impacting potential exposure to lead and childhood lead poisoning, which are some of the things in the CDC SVI. So this is just one way to look at it. And it was interesting, as Joffrey found, to find out that it didn't line up perfectly. So sort of spurs us on to do more work to look for other contributors and other things that may be affecting kids' possible exposure to lead and risk. So hope that helps clarify.
It does. Thank you, Diana.
Great. Well, thank you all for listening to Joffrey's wonderful presentation full of facts and figures, and we greatly appreciate the hard work that you did. We're going to go ahead and take a 2-minute break and to step away from the computer for a half second, and we'll come back at 1 o'clock for our final presentation of the afternoon. So I'll see you all in a few moments. Aloha, everyone. Welcome back. Our next presenter is Carmelita Lobos, excuse me. She will be introduced by her mentor, Lauren Van Hukelum, who is the Senior Response Coordinator at Kaiola and oversees the monitoring of the endangered Hawaiian monk seal population on Hawaii Island. She runs the center's 24-hour hotline for reporting seal sightings around the island and coordinates a team of approximately 125 volunteers. These volunteers conduct research and population assessments on a daily basis. So please join me in welcoming Lauren to the Zoom microphone.
Thank you, Lisa, and hello, everybody. It's my privilege to be introducing Carmelita Villalobos, who was an intern with us during the 2021 year. And she came at a great moment for us, right as the pandemic was kind of in full swing. We actually had to kind of slow down on our volunteer efforts, and Carmelita came in and she was able to help us out as essentially, a staff member who were able to get special permissions to go out and monitor our Hawaiian monk seal populations on the island. So she's done a lot of things for us. She helped out with our education department that went virtual. She's actually helped out with some of our transport of Hawaiian monk seals from our hospital. But the core of what she did with us was through our response program, out in the field, working with other volunteers. And towards the end of her time with us, she actually became a dispatcher as well. So really helping with the efforts around response as we cover all of the coastline on the Big Island for any Hawaiian monk seal that shows up on the beach and is reported. And another great thing that she did with us that was invaluable is she created a Hawaiian monk seal ID sheet. So she basically all of the different identifiers for our Hawaiian monk seals on the island. And I think she's going to talk a lot more about that in her presentation. So without further ado, I'd like to introduce Carmelita.
Thank you, Lauren. Okay, let me get this pulled up. Okay, mahalo again, Lauren, for that introduction. I really appreciate it. And aloha all, and thank you for joining me for my talk today about Hawaiian monk seals and their population on Hawaii Island. I am so honored to be able to wrap up this day of amazing presentations. I am truly proud of all of my classmates, all of whom who have made my success as a professional intern possible. Since last September, I have been working at Kaiola, which is an extension of the Marine Mammal Center, to monitor, protect, and educate others on the importance of Hawaiian monk seals in Hawaii. This has truly been a life-changing experience as I had the chance to work with an endangered species, be around the most passionate volunteers, and receive the guidance of a mentor who helped me realize and begin to achieve my professional goals. I will begin my talk today by speaking about what brought me to Hawaii and where I initially started my internship work at the beginning of this program. I will discuss how this brought me to the various jobs I held during this past year. I will then begin speaking about my current internship with Kaiola by providing background on Hawaiian monk seals, introduce the Marine Mammal Center and Kaiola, and discuss my role there. I will then lay out the objectives of my internship, the approach I took to accomplish my goals, and the deliverables I was able to create. So coming to Hawaii was a bit of a leap of faith for me. And by a bit, I mean, it was a huge jump. All I knew before coming to the island was that I loved the ocean and I wanted to spend my life working to conserve it. So my 21-year-old self convinced me that moving to an island 4,000 miles away from my home was the only way to do it. And so I moved to Hilo. I fell in love with sharks while on a trip to the Bahamas with the Shedd Aquarium in Chicago and was determined to work with them. So on the first day of our internship class, I expressed my interest and Lisa Canale connected me with Hawaii Uncharted Research Collective.
Their mission is to conduct scientific research on whale sharks and merge this with community engagement, which seemed to fit in well with my professional objectives. So for about 6 months, I worked with HERC to develop curriculum for Hawaii schools, grades K through 12. My goals here were to develop interactive and educational activities to teach whale shark biology and the importance of their presence in Hawaiian waters. With this all happening during the start of COVID, the idea was that I would make all of these deliverable through Zoom and be accessible to teachers as a lesson plan that they could complete on their own if needed. Although I have a background in substitute teaching, writing curriculum was not something that I was comfortable with or even had experience doing. But this internship allowed me the creative freedom to explore my inner educator and get comfortable with developing fun content for kids. This experience helped prepare me for the 2 jobs I held during my second year of TCBES. So first I was hired at Imiloa Astronomy Center as a teaching assistant for their Halal Lamaku program, which provided students grades K through 5 with support during the pandemic. We made sure students stuck to their online class schedules and also provided them with place-based activities that allowed them to engage off of their computer screens. After I completed that, I started at the Center for Community Engagement here at Hilo, where myself and 2 other TCBES students wrote and delivered 3 STEM-based activities to middle schoolers via Zoom. Our goal here was to engage middle schoolers in STEM activities that followed next generation science standards to get them excited about Science Olympiad. Overall, my experience with Hawaii Uncharted Research Collective made me a better science communicator and educator, which allowed me to be successful in these 2 positions. Although I enjoy working with students and appreciate the skills I got out of these experiences, I really didn't want education to be the main focus of my career after graduate school. I was looking for a more hands-on internship, which would take me closer to the ocean and help me find where I belonged in the robust field of conservation. And so I wrapped up my internship with HERC in August, and Lisa almost immediately connected me with my current mentor, Lauren, who is the Response and Operation Coordinator at Keikaiola. I was able to start the onboarding process at Keikaiola before the semester even began. This was interesting because Keikaiola was operating in such a different way than it normally does because of the COVID-19 shutdown. For those of you who are unaware, Hawaii shut down all the beach parks, so there were no people on the beaches. Places that were normally packed with people were completely bare. For this reason, volunteers that were usually needed daily to respond to seals and conduct outreach shifted to doing voluntary beach surveys, as they were only needed to conduct visual checks of the seals. Even with this being the case, I was immediately able to become involved in an internship that provided me with hands-on experience working in the field of conservation. Before I get into what I did, I just want to give some background on Hawaiian monk seals. Hawaiian monk seals are one of the most endangered seals worldwide and are the most endangered pinniped found in US waters. They are one of only 2 monk seal species, the other of which I believe is found in the Caribbean. Although endemic to the Hawaiian archipelago, many people have the misconception that they were introduced and are considered a nuisance by fishers and beachgoers alike. Hawaiian monk seals were around before the arrival of the Polynesians in numbers upwards of 15,000, but upon their arrival, they were hunted and killed to near extinction. Hawaiian monk seals were extirpated early, so people did not grow up with them around the island, and they were misinformed when they started to be present in the main Hawaiian Islands. Misconceptions and poor outreach led many to hate them, even going as far as killing them for no reason. The result is that today there are only 1,400 monk seals left in the entire Hawaiian archipelago, 300 being found in the main Hawaiian Islands and only 10 being found on Hawaii Island. Hawaiian monk seals are named as such because of the way the skin on their necks folds like a monk's hood. They are born black, but get dark gray to brown on their dorsal side and light gray to yellowish brown on their ventral sides as adults. So this is a mom next to her pup, which really shows the contrast in color and size. Full-grown adults weigh anywhere between 400 and 600 pounds and can live to be between 25 and 30 years old. Hawaiian monk seals spend their time between the terrestrial and marine environments, with 2/3 of that time being at sea. In the ocean, seals can be found foraging or traveling and rest molt and pup while on land. This number kind of shocked me when I first found it out because as a volunteer, we spend so much time looking for these seals on land, and it seems like they're always there. The time spent on land is vital to their survival and is why the work of Ke Kaiola volunteers is always needed. During their time on the terrestrial environment, protection is needed so that the public respects these behaviors. I will now dive into these behaviors a little bit further. So when I encounter a monk seal, this is usually what they look like. Most of the time with monk seals, my time is honestly spent watching them sleep. So one of the most frequent questions I get asked is how long they'll rest, and it honestly depends on the day and on the seal. Time on the beach can range anywhere from a few minutes to an entire day. I also get asked if they like to hang out in the same spots. And I will say that each seal has its own distinct personality and has beach preferences just like you and I do. Monk seals are mostly solitary and are almost always found alone. But I used to say that they were always found alone, but of course I had to eat my words just a few weeks ago when I said that while I was out on response, and then 2 seals rolled up on the beach together. Anyways, for this reason, even though there are only 10 seals on Hawaii Island, a very large volunteer force is needed to monitor them since they are rarely found in groups, meaning that if we have 4 seals hauled out in a day, it usually means that they are in 4 different locations and require 4 different volunteers per shift. Similar to the resting behavior, every year monk seals undergo a catastrophic molt where they shed their top layer of fur and skin. This takes about 1 to 2 weeks, depending on the seal's age, weight, gender, and female's reproductive status. This is done on land, and again, the seals are mostly sleeping and scratching off their old fur. Seals won't return to the water until they are completely finished, or will do so only briefly. Lastly, female monk seals give birth or pup on land. Female monk seals can start reproducing as early as age 5 and usually do so until around the age of 10. Gestation takes around 10 to 11 months, and during this time, mothers put on extra weight to be able to support their pups before weaning. Pups are born at around 25 to 30 pounds and are weaned between 110 and 175 pounds. This means that in a short 5 to 7 weeks, pups gain an impressive amount of weight, which they gain solely from their mother's milk. Mothers do not return to sea to eat during this time and lose weight while the pup gains it. Once weaned, monk seals are generalist feeders, meaning that they eat anything from fish to squid, octopuses, eels, and crustaceans. They forage at the sea, in the sea, mostly at or near the sea floor, which is when their diving ability comes in handy. So they can hold their breaths for up to 20 minutes and dive as deep as 1,800 feet. Generally, though, they take dives around 6 minutes to 200 feet. Although it is thought by some that monk seals create competition for fishers, they don't actually target locally popular game fish. Monk seals also travel in the water. They don't complete a seasonal migration, but can be known to travel hundreds of miles up and down the Hawaiian archipelago. It is not uncommon for seals to hang out around one island all year and then travel to another island to pup. For example, we have a seal who hangs around Hawaii Island for most of the year, but then travels to Kauai to pup, and she actually just did this a few weeks ago. And I included this picture even though it's blurry because there is a monk seal right there in the water. Sadly, there are many threats to the Hawaiian monk seal population, and unsurprisingly, the threats vary by area along the archipelago. In the northwestern Hawaiian Islands, entanglement is by far the largest threat. Derelict fishing gear is commonly found in those waters, and monk seals, being the curious creatures they are, often find themselves entangled in gear. Food limitations due to overfishing are also a threat, in addition to shark predation on pups. In the main Hawaiian Islands, fisheries interactions, human interaction, and infectious disease are the top threats to Hawaiian monk seals, and I will discuss these in further detail. Hawaiian monk seals have the highest entanglement rate of any other pinnipeds. In the northwestern Hawaiian Islands alone, 800 metric tons of debris have been removed since 1996, and I don't even think that's the most up-to-date number. Although this is less common on the main Hawaiian Islands, we still see this occurring along with increased hookings due to recreational fishing. This is our youngest seal on the island, and you can see in this photo that he has 2 rings around his neck, which is most likely the result of some kind of interaction with marine debris. Fortunately, he shed whatever it was on his own, and it didn't seem to affect his health at all. More likely a threat to seals on the main Hawaiian Islands is recreational fishing. Seals are curious and also aren't afraid of helping themselves to a free snack. So this brings up the issue of unintentional hookings. We try to encourage fishers to pay attention while they are fishing and bring up their line for a few minutes if a monk seal is in the area. We also encourage the use of barbless hooks to decrease the harm done to seals if they are to be accidentally hooked. A barbless circle hook is a regular circle hook with a flattened barb. Research has shown that barbless circle hooks work just as well as barbed hooks, but because they are easier to remove or are able to be self-shed, they minimize impacts on protected species such as monk seals. We usually don't try to remove hooks from seals and simply monitor them to make sure that they lose them on their own and do not develop an infection. As mentioned above, the seals and humans share beach spaces. So with an increasing human population on Hawaii Island, habitat competition has become an issue for seals trying to access beaches. Overcrowded beaches make it difficult for seals to haul out, and when they do, they are often met with human interaction. Disturbance such as people getting too close changes the way that the seals behave, which isn't good for their health. I really like this picture because it seems like a harmless picture of a resting monk seal, but there are many things wrong with it. So this picture was taken from way too close of a distance. You can see that the animal is pretty aware of the cameras looking right at it. The simple act of grabbing the seal's attention is disrupting its normal behavior. I actually responded to this seal and almost stepped on her because the person taking me to her did not understand how far away we should be from her, and I didn't see her. There have been instances where people will go as far as killing seals to prevent them from eating fish, and in instances for no apparent reason at all. Feeding is also an issue in the main Hawaiian Islands, but to my knowledge, it is not a huge issue on Hawaii Island. So the last big threat on the main Hawaiian Islands is infectious disease. If you live on Hawaii Island, you may be aware of the large feral cat population. Cats spread toxoplasmosis, which can be deadly to Hawaiian monk seals, as there is currently no treatment or cure. Monk seals may not directly interact with cats, but they can be exposed to the same beach areas, making them vulnerable to the disease. It is clear that the Hawaiian monk seals have faced numerous stressors which have relentlessly threatened their population numbers. Luckily, the Marine Mammal Center, or TMMC, has been working to combat these stressors since it opened its doors in 1975. To date, the Marine Mammal Center has rescued more than 2,400 marine mammals throughout the California coastline and the Hawaiian Island chain. TMMC has been a trailblazer when it comes to ocean conservation, as it has been focusing on marine mammal rescue, research, outreach, and veterinary medicine for decades. The ocean is in need of our help because it is suffering from a multitude of anthropogenic threats, including marine debris, overfishing, and rising temperatures, each of which have threatened the livelihood of the organisms that call the ocean home, including Hawaiian monk seals. The main mission of the Marine Mammal Center is to advance global ocean conservation through rescue and rehabilitation, scientific research, and education to combat the negative effects of the changing ocean on marine life. All of this work is possible because of the more than 1,300 volunteers that donate their time to responding to the animals and conducting public outreach efforts. In July of 2014, the Marine Mammal Center opened a state-of-the-art hospital and visitor center on Hawaii Island named Keikaiola, or the Healing Sea. This facility was built to help save the Hawaiian monk seal population and is the only facility in the world of its kind. Keikaiola is equipped with medical facilities and rehabilitation areas for sick and injured seals. Of the approximately 1,400 monk seals in the wild today, it is estimated that nearly 30% are alive directly because of the conservation efforts by NOAA and the Marine Mammal Center. Seals all along the Hawaiian archipelago that need care are sent to Keikaiola for rehabilitation, since it is the only facility built specifically with Hawaiian monk seals in mind. The work of the dedicated staff members and volunteers make Keikaiola a leader in animal care and an asset to the Hawaiian monk seal population throughout the Hawaiian archipelago. In addition to animal care and rehabilitation, Keikaiola has around 50 active outreach volunteers that dedicate their time to protecting the Hawaiian monk seals out in the wild. The Marine Mammal Center supports a monk seal hotline, which takes calls every day regarding Hawaiian monk seals. TMMC relies on the public to report monk seal sightings since our seals are tagged but not tracked. My role at the Marine Mammal Center is Hawaiian monk seal conservation response intern. For about 8 months, I worked with Lauren to gain hands-on experience working in response and outreach. In the beginning of my internship, which again was during the COVID-19 lockdown, there were no people on the beaches and really no reason to respond to seals. Lauren was therefore able to bring me around to different beaches on the west side of the island and taught me how to access the different locations, showed me where seals like to haul out at, and gave me tips on how to identify the different seals. Even though response wasn't necessarily needed, it was still vital to monitor the seals to make sure that they were healthy. As I got more comfortable with the beach locations and with identifying seals, and as COVID-19 restrictions began to lift and people were returning to the beaches, I was able to begin my role more fully as an intern. My responsibilities at the Marine Mammal Center began to include response and outreach, dispatch, volunteer training, and then creating deliverables to assist with seal identification. At the beginning of my internship, I was able to work 2 shifts per week, and it mostly worked out that I would shadow Lauren for one of those shifts and then be on my own the next day. Volunteer shifts at the Marine Mammal Center go from 8 a.m. to 6 p.m. and are split up into 3 sections, so shifts lasted for 4 hours each. In the morning, Lauren would send out a text telling each volunteer where to go, depending on if people called to report seals that day or not. I would then hop in the truck and head to wherever I was needed for the day, or if there were no calls, I would head to beaches that we frequently see seals at to survey the area for them. Once a seal was located, hauled out on a beach, I would start my response. With the number of people on the beaches nearly back to pre-COVID times, I would begin by assessing whether a seal awareness zone, or SAS, was needed. A seal awareness zone designates a 50-foot perimeter around the seal on all sides, the goal of which is to give the seal space so as to not disturb it. You can imagine that this is a very large area because each side of the seal needs to have this space. And I swear our seals love to cause a commotion, so they seem to always pick high traffic areas to haul out on. A lot of the time we find seals already plopped on the beach in the morning, but there are many instances where seals will haul out in the middle of the day when there are a ton of people on the beach already. And it is my job to clear the area and set up the SAS. This means grabbing signs such as the one shown on the slide, which relay our message, and rope to prevent people from accidentally walking into the seal. So this is a better example of a seal awareness zone that Lauren and I set up together. The seal is clearly resting and is unbothered by our presence, which he's he seems not to notice us at all. My next step here would be to identify the seal. So to ID seals, we look for any bleach marks, scars, or tags that are unique to the individual. The seals born on Hawaii Island are tagged with bright orange tags, which can be read with the help of binoculars, but scars and marks are also extremely helpful to aid in ID. Once the seal is identified, the information along with any visual health assessments we have made is reported back to Lauren on the response phone. When all that is said and done, I more than likely have a crowd already ready with questions for me. Outreach is a huge part of my internship and is so important to the success of TMMC. It is during outreach that I am able to educate the public on the importance of Hawaiian monk seals and their conservation, tell them what they can do to help seals, and answer any questions that they may have. My goal is to have a positive interaction with the public every conversation I have, which has taught me how to listen to people and mold my conversations around what they want to get out of the interaction. In addition to this, it is important for me to stay with the seal to make sure that people are aware of it. You may think that people will read the signs and stay back, but there have been many instances almost every day where people are simply not paying attention and walk right through the signs. More often though, people just love to learn about our seals, so I am there to talk story with them and answer any questions they may have. By doing this, I ensure that beach patrons keep their distance so as to not disturb the seal, and I also get to educate the public on monk seals. Although it is my top priority to protect the seals, I really want to make sure that people have a positive experience with me so that they learn to care about the seals just as much as I do. As schoolwork picked up and I went down to only one shift per week, I took on additional responsibilities at this time. I started running the response phone on Saturdays, which has strengthened my customer service and communication skills. People call or text us at 808-987-0765 when they spot a monk seal. I field these calls, gather information from the caller, and decide if a volunteer is needed for a response or not. Since I live in Hilo and can't easily access Cape Kaiola to pick up the dispatch phone, we started using an app called Google Voice. What this app does is forward all calls that come into the actual response phone directly to my phone, so I'm able to take calls from anywhere. I also use this app to start the volunteer text thread in the morning and keep in touch with volunteers throughout the day. After my day of dispatch and response, I enter all of the monk seal sightings from the day into our dispatcher data entry sheet. This allows us to keep track of where seals have been and helps us monitor their health. This is accessible to NOAA as well in case they need information on our seals. All these responsibilities have prepared me for one of my favorite parts of my internship, which is helping to train new volunteers. By the time volunteers get to me, they have already gone through a few training sessions with Lauren, so she really does all the heavy lifting. And I get to do the fun part and show our new volunteers around to different beaches and expose them to outreach. I always find myself taking the initiative to conduct outreach when I'm out with volunteers, and I'm always completely surprised when they take control of the conversation and just know so much about these seals. This is one of my trainees here, and as you can see, I'm totally nowhere near him taking the picture because he had everything under control. The many volunteers I have worked with have taught me so much about seals, outreach, and conservation, and it has been really great to work with people in a time when we have all had limited human interaction. For the last part of my internship, I created a new seal ID PDF to assist volunteers with identifying seals. So my goal here was to point out every scar, mark, and tag to help volunteers easily identify our seals out in the field. For each of the 10 seals, I provided a page of background information, which may include birth dates, any pups or siblings they may have, where they like to hang out. And then the second page was dedicated to pointing out all of their unique physical characteristics, which meant spending hours going through the Cape Kaiola Dropbox account with years worth of pictures in it to find the best pictures of each mark on each seal that I was trying to point out. This really helped me get to know the seals and become an expert in identifying them. This PowerPoint is a working document, so it can be edited whenever, even when I'm done. New seals can be easily added to it, and old ones can get updated if they get something like a new scar, for example. As an unexpected bonus, I got to assist with the transport of 2 seals back to their home islands of Oahu and Kauai. Both seals were being rehabilitated in the hospital for around a month, and once they were ready to be released into the wild, I assisted with loading the seals in their crates here and onto the Keikaiola trucks. We then drove them to the airport to be transported to their final destinations by the US Coast Guard. The purpose of my internship with the Marine Mammal Center was to assist in monitoring the Hawaiian monk seal population on Hawaii Island and to engage the community to educate them on monk seal conservation. While doing this, I gained hands-on experience working in management and volunteer operations by operating the monk seal response hotline and organizing volunteers, and also gained exposure to working with an endangered animal. I am very thankful for my mentor, Lauren, for showing me how to gracefully work with the public and for showing me how to manage a volunteer force. I want to mahalo her for all her valuable guidance and time in helping guide my journey in becoming a professional in the field of conservation. I also want to mahalo my cohort for being there for me constantly, all KKO volunteers for sharing their knowledge with me, and Lisa Canale for being so supportive and understanding always. With that, I'll end by giving you the monk seal hotline number one more time so you have it. It is 808-987-0765. Unfortunately, Saturday will be the last time that I will be answering the phone, but I promise that Lauren will answer for as long as there are monk seals on Hawaii Island. Mahalo.
Oh, fantastic. Thank you so much, Carmelita. That was just marvelous. I think my new favorite species is the Hawaiian monk seal, so thank you.
Yes.
So I want to invite everybody to either type a question in the chat, or if you're brave, you can just go ahead and unmute yourself. And while we're waiting for the first question from the audience, do you have a favorite seal? I would think that you might. I'm just wondering if you do.
I do. Lauren just asked me this a few weeks ago, and I was like, oh my gosh, I do. It's Manuiva. She's a little girl. She's so cute. She just has the most beautiful coat, and she's always just like glistening in the sun.
And then can you explain a little bit about what bleach marks are?
Yeah, I don't necessarily know why they occur, but I can compare them to like a birthmark. They're just born with them sometimes, so it's just like a discoloration of their skin.
All right, audience, does anybody have a question? Oh, here's one from Dawn. She says, do monk seals eat during their molt?
To my knowledge, they don't. So they're on the beach for like 1 to 2 weeks. And again, to my knowledge, they just return to the water if they're like too hot and need to cool off a little, because it's a pretty like uncomfortable experience for them. So they're kind of just hanging out on the beach until they're done. But Lauren can correct me if I'm wrong on that.
No, you're good. They might passively eat. You know, if an octopus happens to go past them when they're in the water, they're not going to pass it up. But yeah, they don't act, as far as we know, they don't actively eat. But my question, Carmelita, is, you know, you mentioned that you had a little bit of experience kind of with the course curriculum area, and then you came to us and you worked more out in the field in conservation. So do you feel going forward into the future, do you have a better idea of what you want to do with your future career after this program?
That's a great question. If anything, I feel like I have just a robust skill set now where I actually feel qualified to do things now. So I feel ready to find what my next step is. And before I wasn't even there. I could not imagine myself working in the field of conservation, even though it's what I loved. And I feel like I'm there now because of all of these combined experiences.
So let's check in on to our live stream. Becky, do we have any comments or questions over there? I think our live streamers are very shy, or maybe they know how to reach you, Carmelita, with their questions later. All right.
I have a question.
Oh, sure. Please go ahead.
Yeah. Hi, it's Brianna.
Hey, Bri.
I was wondering, like, how long do the safety zones like typically stay up for?
Oh, that's a good question. So it depends on how long the seal is going to be on the beach, right? So we could set up the zone around them and then they decide they don't want to be anymore and they leave and then we'll take it down. But if they end up staying for an extensive period of time and it gets to around, I think about 6:30, so when the sun's setting, most of the beaches here close at 7. So we can take down all of the signs and ropes and put it away because the seal will be fine on the beach because there will be no more people there. So if they stay, we keep it up until the night.
Okay, thanks.
So there's a question in the chat, and the question is from Morgan, and the question is, are monk seals usually alone when you find them? That's the question.
Yes. So usually they're alone. I used to say always, but that's not true. Sometimes they'll haul out together or you can see them playing in the water together sometimes if it's our younger seals. But mostly we do find them alone on the beach. And then I see that Doe's asking what the Hawaiian name for the monk seal is. And I honestly can't answer that. Lauren, sorry to put you on the spot again. I don't know if you can.
Yeah, I can do it, but I will apologize. I'm going to butcher this. There's actually a really great YouTube, if you look it up, where little kids will sing it to you. And it's Ilio Holo Ika Ua Ua is the Hawaiian name, and it basically means dog running in rough waters. So a lot of times Hawaiian monk seals are likened to dogs because they're very curious by nature, which is another reason that we set up these perimeters around them, is we don't want them to become used to humans. We've had issues in the past where, you can imagine a pup is 25, 30 pounds, super cute. People are like, oh, sure, come play with me. But once that pup becomes a 600-pound adult seal, that's a whole different story when it's trying to hug you and play with you. So we've actually had to, not us personally, but there have been times in the past where they've been relocated because they've become too ingrained in a community and actually have, there's been some health concerns there.
Thanks, Lauren. Mm-hmm. And I see that there's a question, what can a monk seal do if a person gets too close to them? And also that Katie asked me to do my best seal impression, which I feel like they're related because if you get too close to them, they will bark at you if they don't want you near them, which they usually don't. So in that way, again, they sound like dogs with their barks. So yeah, they'll be defensive if they feel threatened.
All right. Well, I think we're all waiting for your impression, but if you want to be shy, that's okay. We can move on to the next part of our talk. What would you like to do? You want to do it or you want to be shy?
No, oh my God.
Okay, all right.
Maybe on graduation day.
The after presentation. You'll have to see it in person. All right. Well, I want to give you all a huge thank you for being with us this afternoon and morning. It's been fantastic, and a big shout out to our 6 presenters today. The talks were marvelous. They will be housed on the TCBES YouTube channel forever and ever. So please tell all your friends. And what I'd like to do now is hand over the floor to our graduate students for a closing ole.
So we just wanted to offer up a mele, oli mahalo, to express our gratitude to Lisa Canali, all of our mentors and professors over these past few years. Uhola ia kamakalo ala. Ua ike alohala. Uka ia kahalo. Pavehi maina leua. Mai ka ho'oku ia kahalava ila. Mahalo e na apua. Mahalo e na punala ea. Mahalo me ke alohala. Mahalo me ke alohala. Mahalo.
Thank you, everybody. Thank you so much. If you're brave, please unmute yourself and give them a big shout out, a big mahalo. And I'd like to invite you all to attend graduation, Zoom graduation again, and it'll be in 8 days, and the email will be going out soon. So 1, 2, 3, unmute. Mahalo, good job.
Congratulations. Good job, everybody.
Congrats. Thanks, everyone. Congratulations to everyone.
Yeah. Thank you, everyone.
All right.
Woo.